disciplinarian.
All right, thank you for returning from lunch. And a special thanks to the phone people for returning. Can the people on the phone hear? Yes, I'm so sorry about the beeping problem and the need to cut you off. So we have a new phone number, and I think we're good to go. All right. So we are going to do just a small change in the agenda. There was some lingering questions that people had about the passage section and passage speakers in particular. So we were going to dedicate about 5 or 10 minutes to if people had additional questions for Sean or Nick or Damon or Ralph. Oh, Ralph, you're here. Yay. Okay, so we have everyone here except for Damon. Hopefully Damon will come in if people have questions for him. But if anybody, so does anybody have questions for any of the passage session speakers from this morning? We just didn't feel like we didn't get enough time to quite finish that. Okay, so Josh is going. Yeah, if you guys want to come up, that would be great.
Sure.
Yeah, let's make use of this, the panel here.
Yeah.
Test, test, test. Hello?
Yeah, there's no. Brian's walking around. Got it? Oh, you're not going to give me the microphone?
Yesterday, we heard from one of our tribal leaders, Mr. Bruce Jim. I don't know if Bruce is here this morning, but one of the things that he brought to our attention again, as he has several times now, is this idea that sound from the water creates a turbulence, and it's probably something that the lamprey can hear and would be an attraction to maybe the devices that you're building. And I wonder if you folks might have any comments, speculation, thoughts about how and where we might be able to use some sort of a facility like that, and how we might actually be able to evaluate it.
I'll start. I think that's definitely a good point. You know, I definitely think they key into the sound, you know, the splashing sound of water, and that's basically what the vertical wetted wall tries to create, or even LPSs, and but how you do that at a large scale or with a bigger impact, that's something we have to explore further.
I would say the same thing about the, who knows how to separate the effects, but I would suspect that might be a factor with the LPSs and the wetted wall, which I should have mentioned that at Bradford Island Ladder at Bonneville Dam this coming winter we'll be installing a wetted wall structure as well. And Bruce's concept is duly noted, and I know Brian McElwraith and I had talked about that same concept that he mentioned yesterday a few times. I'll also say that walking around the dam during the passage season and talking with our project biologists that are out on the concrete, we definitely see examples of where, you know, for example, the LPS outfall chute at the Washington Shore ladder, some of the water from that kind of splashes on the opposite wall in the exit section of the fishway, and folks have noted seeing some lamprey kind of start to climb the lower part of that wall. So I mean, we definitely see that kind of attraction to splashing.
That's the first time I've heard of that theory, and I don't know. I'd be interested to hear of other people's ideas on ways to test that. I guess my idea would be is that it would be challenging to try and take advantage of that in areas in and around a dam, just because there's so many competing, you know, acoustic interferences. So in some specific applications, I can see that if there is an attraction there, it could be taken advantage of. But in a lot of cases, I think it could be challenging just because of all the competing interferences.
And I would add, we hold lots of adults at our tanks, and every time it splashes on the wall, that's when you see adults climbing up, and so we see it all the time. And also in my bathtub, in my new house, I have a big bathtub, and I sometimes kind of pour water, climb under the pool, and you can hear those sound pretty
You should
loud.
stop there.
It's pretty loud. Yeah. How much you can hear that.
Sorry, won the award, Ralph.
We got a question over here.
Yes, I'm wondering why we haven't really talked about fluctuation, because the dams, they raise and lower that water so much, and there's a lot of exposure that sometimes the areas that we know there should be amicetes is dry. So to me, I'm thinking from my past experiences of watching companies pay a fine and continue business, and I hope this isn't just a Band-Aid approach to recovering these animals. And so I hope that I can get an answer. When are we going to start addressing that issue of the level? Because there's other interests too. I don't hear anything about the irrigation. I don't hear anything about the navigation. Because when those smolts and other species come down, the waves of those barges do strand some species. So there's a lot more, and we need to really address that issue of the drastic lowering. Because there's areas that are exposed, sandbars, where they are. And you should go to some areas where the water, how it comes down off the hillside, and you will see how it is, the rocks is, because down there at Fisher Hill Bridge, we used to have a lot of the Lampre in there until the farmers up above took all of the water. The same in Swale Creek up at Wakaikas. There used to be a lot of them in there. But the farmers in Centerville, again, cut off the water. So we need to address the whole issues, not just with a Band-Aid approach. So I hope that we can get an answer from Sean about how are you making sure that these amicetes won't get stranded and done away with.
Thanks, Wilbur. A couple things on that. I share your concerns, and one of the things I didn't touch on because my talk was focused on the adults was we did actually a few of the folks in the room here. Bob Mueller and his group. I think Joe Skaliski is in here, and Marty Litke. Helped us look at, or at least doing a preliminary look at the potential kind of effects of the hydrosystem operations on those shallow kind of rearing areas in the main stem, just to get a sense of the risk. And it was kind of a series of both going out and looking at distribution using a deep water electrofishing gear that Fish and Wildlife Service has, and then USGS did some simulations to see how the amicetes respond to dewatering. It was just kind of a first look, but I think folks that were working on it would agree that there's definitely that concern as to the particulars of the dam operations and how they might be affecting fish. I don't think I know enough both about the operations or the fish to have a definitive answer, but I just want you to know that it's something we're thinking about and working on. On navigation, I should make a plug that the Corps the Portland District just kicked off environmental impact statement EIS process, kind of the scoping process for our lower Columbia River main stem navigation channel maintenance. It's a big, huge, multi-year process to look at the effects of that program, and also how we manage sediment. We did a site visit recently. A few folks in the room were on a site visit where we actually got on the Dredge Oregon to look at about how the dredge operated and an upland disposal site, and about half the group were Lampre folks from the Lampre Technical Work Group are thinking about the effects of dredging. So it was really good to see that Lampre were on the radar for the folks that are working on that big EIS. I'll just say in the last few years, just internal to the Corps, I've been excited, not just with that project, but with other projects, to see folks thinking about it, not just in terms of passage, but dredging, our regulatory program and how we issue permits, and some of the other operations that we have. Hopefully that at least partially addresses your question, that it's being heard and the concerns are being heard.
All right, thank you, Sean. Puts my mind at ease.
Good.
Josh, did you have one? Yeah, maybe one more, and then we're gonna continue.
I'll hold it. Yank it away from me. So the one thing I haven't heard folks talk about is this 2017 return is probably about 30 times the number of fish that we saw in the dismal 2010 return. It's been a tremendous year-over-year increase, and I haven't heard a lot of discussion as to why folks might think that is occurring.
I always ask you about those kinds of questions.
I would assume that there is some sort of ocean effect there, that there's something going on in the ocean that's improved macro to adult returns, but I also like to think, at least in the Clackamas, a part of it is our improvements there, but you know, it seems to be that particularly in 2017 there was just regional uptick, so that in my mind that there's got to be an ocean effect there.
Like, I think we heard about the Pacific hake fishery really improving, and like, I think those may have a big impact on Lampre. But in the Yakima, those numbers that I showed, those were from the 2016 rootstock, so those were, you know, the ones that were coming in 16 and migrating in the spring of 17, and we had a huge increase there that wasn't seen in other places, so we hope that trend continues this year.
Okay, thank you guys. Oh, yeah. Brian's gonna make an announcement.
During Ralph's speed presentation, I think maybe he mentioned his ID, the updated ID guide, front and back. And so we do have 79 copies of that. So if you're from Idaho, definitely take one. If you're from California, definitely take one. If you are from Washington and Oregon, maybe take one. If you know Ralph or I, don't take one. We just want to make sure the folks that don't come around very often have a chance to get one, and they're on the table where you signed in. And if you haven't signed in, make sure that you do, because I want the biggest meeting at CRITFC to be a Lampre meeting, and if I can have some data to support that, that would be really cool. So make sure you sign in, and you can use a fake name, that's fine, just make sure you're on there.
Bob, did we locate these?
Not that I'm aware of, but people do need to know that we do have those books that Mr. Don Crandall helped put together with our good friends from the Met House, and those will be available at some point, and so hopefully we can find those. The box, anybody know where that box went to?
We had a box of a bunch of these yesterday, and they didn't make it over here, but we will try and find them. As Bob said, it's a...
Wants other copies, get a hold of me. I don't know exactly how to do that, but I have more copies. mind thinking, because we have a lot more funding going to salmonid-based restoration, to be thinking about lamprey from the early planning stages of salmonid-based projects. So there's a lot of information in the guide, but there's also, it was built to have a lot of resources and people you could contact, and because it's never, you know, we're learning a lot right now about lamprey, so that's really the intention. So get a hold of me or Christina, and we can direct you to more guides, but the thought is to get it out to the folks in the salmon world as well, and they're really important because there's a lot more of that specific restoration going on.
Thanks.
It is electronic, and it's on the Methouse Salmon Recovery Conservation website, which is www.methousesalmon.org, and there's a PDF posted on that website. So it is. Yep.
And we can probably put a link to it if you want on our Fish and Wildlife Service page then. Okay. All right, so just a couple things. We're going to cut short the break a little bit, just so you know, just so you're aware of that. If you need to leave, you know, during the sessions, please go throughout the back door. But just to kind of catch up on time, we are going to, just so you know, we're going to cut the break a little bit short. And if you're speaking in this one, we're having an issue with the sound in the back. So when you're speaking, try and speak this way instead of this way, like, you know, with your presentations, just for the upcoming speakers. And if you do feel like you're going to roam around a lot, you can, you know, if you want to be back here talking, maybe use the microphone. This microphone doesn't work the greatest. So just try and be cognizant of trying to speak this way so people can hear in the back. Yeah? Okay. Anne, take it away.
So my name is Ann Broda. I work with U.S. Fish and Wildlife Service at the Mid-Columbia River Fisheries Resource Office. And I'm really excited to see such fantastic turnout here today. I'm especially happy that everyone came back after lunch. And we're going to be talking about a really interesting topic, a novel topic here in this next session, which is environmental DNA, or eDNA. So the sort of schedule of this session is that we have 3 speakers, each of whom are going to be speaking for about 20 minutes. And after each of their talks, you kind of have built in a 5-minute question period. And then we're hoping at the end, once all the speakers have gone through, that we'll have like a 10 or 15-minute time for a general discussion for any outstanding questions or anything that got missed in those shorter question sessions. The kind of goals of the session today were really to provide some background on environmental DNA. So what it is, how does it work, what are some of the sort of lamprey-specific considerations for doing eDNA work? What are the strengths and weaknesses of this approach? And also to provide some familiarity with specific applications that eDNA is being used for in lamprey conservation right now. So I don't want to take up too much of our limited time, but I want to go ahead and introduce our first speaker, who is Kelly Kareem. Kelly is an aquatic resource and research biologist at the National Center for Genomics and Fish and Wildlife Conservation. And as soon as I cue up her talk here, and she's going to be talking about recent applications of eDNA. As soon as I get it. All right, Kelly.
Great. Thanks, Anne. So Anne mentioned that I'm talking about applications, and I think that may be a little bit of a lie, because as I was putting this presentation together, I realized that really what I wanted to do is to provide a lot of background to all of you so that you can start to really gain a good understanding for the type of information and interpretation of the information that both Carl and Marty are going to present to you next. So I'm going to kind of give like a brief overview of what eDNA is and how it really works as a tool. This is kind of I tried to condense it down and make it pretty accessible, but eDNA has a lot of nuances to it. So if any of you are planning to come to Oregon AFS this year in March, I'll be leading the continuing education on eDNA, continuing education course. And so if you want to learn more in depth and you're planning to come to that meeting, it would be great to have you in that continuing education course. But I'll just move on from there. So what is environmental DNA? It is simply DNA released from an organism into the surrounding environment. And now this could be anything from feces, skin cells, mucus, gametes, etc. But the really great thing about it is that we can collect this DNA and bring it back to the lab and analyze it to understand if a particular species is present. So it seems like this is a really novel tool. And to us it is, but the process of using genetic information to answer questions is really not that new. So for example, we've been using DNA evidence collected from the surrounding environment in criminal court cases since 1986. But it wasn't until 2008 when the first peer-reviewed publication came out using environmental DNA information in the context of wildlife management. And this was that paper, Fisitola et al., 2008. And the researchers were looking at the invasions of American bullfrogs in France and using environmental DNA. And what they conclude is that this technique was able to discriminate between species absence and presence even at low densities. And that low densities is key. That's where eDNA really starts to outperform traditional sampling methods. So this is kind of in the context of wildlife management, eDNA is actually really pretty new. The first publication was in 2008, and here we are in 2017, and you can see that we've had this exponential expansion of publications on this topic. This figure is showing the number of papers published over the course of the last 8 years. And this figure was sourced from one of my colleagues, Dr. Taylor Wilcox. This website is up on there, and you'll see this slide a couple more times too throughout the talk if you want to write it down. But he has a really great, living bibliography of eDNA publications, and so he tries to collect every week, see if there's been a new publication, add it to the list, and sometimes he even provides a little short summary of what that paper covered. So a really great resource there. But you'll notice that in the early years of eDNA in sort of wildlife management, we didn't have a lot of publications. And that's because this first generation of eDNA papers were really focused on the basic research. So helping us understand this tool, what its limitations are, and how we can really best use it. And so today I'm going to focus on kind of a summary of this tool and what we learned in some of these early papers. So the first question that we were all wondering is, how does eDNA compare to traditional sampling methods? Is this another tool in the toolbox, and when will it outperform, or maybe not outperform, some of those traditional sampling methods? This is one of my favorite examples addressing that question. It's another paper from France looking at American bullfrog invasions. And the authors compared traditional sampling methods, which included auditory surveys during the breeding season, as well as visual surveys for egg masses in ponds. And using those methods, they detected bullfrogs at 7 sites. Now, when they looked at environmental DNA detection, they had more than 5 times the number of sites that were positive for American bullfrog. So this was really eye-opening to them. They had vastly underestimated the spread of this invasion, and they concluded, and this conclusion has been reiterated time and time again in published literature across taxa and across systems, that eDNA-based methods are more sensitive, they're more accurate, and more time and cost efficient than traditional sampling methods. And again, it's really key at those low densities where it becomes difficult to actually get an animal in hand. So now that we know eDNA is this really effective tool, what are the best methods for sample collection? There have been a number of publications outlining some guidelines, and here are a couple, one from USGS and one from my working group at the National Genomics Center. And all of these protocols have various spins, and they differ primarily on the type of water body you're sampling or the type of animal that you're looking for. But the thing that they all have in common is that they're looking to maximize the probability of detection while minimizing contamination risk. So if you think about it, even a single copy of DNA can cause contamination, and that's going to result in a false positive detection. So you're going to say that an animal is present when it's not really there. At the same time, if you're properly collecting your sample and you properly collect a single copy of DNA, that could be huge as far as saying, yes, that animal is present here, and so we need to account for it. So we need to be really careful with these two sort of considerations in our protocols. in order to, again, maximize detection, true detections, and minimize false detections. So to do that, all of these protocols take precautions to avoid contamination. So for example, in these photos, you see that the individual collecting the sample is using sterile gloves. We're going to handle the filter with sterile forceps and store the sample properly. And by storing the sample properly, the protocols also emphasized filtering and filtering water in the field. Taking that filter in our protocol, we use silica desiccant, which you see in the lower right-hand corner. We put the filter in some silica desiccant that sucks the water out of the filter and preserves the DNA, and that helps us preserve the DNA in the transit time back to the lab so that we're not having any degradation of those few copies of DNA that might be really critical for addressing our questions. All right, so that's kind of an overview of sampling protocols, and there are many available online. Again, you can find them on Taylor's website. But once you have your sample, how do you actually detect DNA in that sample back in the lab? And now this is where things get kind of nitty-gritty with DNA, but I've tried really hard to break it down so you can kind of understand the concept. Currently what we use are species-specific markers to identify the DNA of the animal that we care about that's like in a sea of non-targets. So for example here we have a bull trout. We're looking for bull trout and we have some bull trout DNA in our sample that is floating in, mixed in with DNA from all the other animals that might be, or that are in the system where that sample was collected. So we want to be able to pull that piece of DNA out and say, yes, we found bull trout DNA, bull trout were present. Now, how we currently do this is with a process called quantitative PCR. It's a highly sensitive process that allows us not only to detect a very small amount of DNA, it also allows us to quantify the amount of DNA in that sample. Currently, this is the most reliable and the most commonly used method for eDNA sample analysis. There are several other labs, including the National Genomics Center, working on other methods that can improve efficiency as far as how many species you can detect in a single analysis. But for now, this single species approach, so asking our lamprey here or our bull trout here, the single species approach is the most common. This is kind of our bread and butter of eDNA right now. And this is a little bit about how it works. So we have a species-specific marker that is an exact match exact genetic complement to our DNA of interest. And so we have all of our DNA in our sample and we want to be able to pull out just this bull trout DNA. So we put all of this into the DNA and our marker into a qPCR, quantitative PCR machine, and we run a quantitative PCR reaction. And what happens when we perform that reaction is that the DNA will float around, the marker will float around, and when the marker comes into contact with the target DNA, you have an exact complement. There's a very strong bond, and what results in the process is the emission of light. So we maximize that emission of light and visualize it. That's what the quantitative PCR machine does. And this is what the output from the machine looks like. In this figure, there are 3 samples. The first one, we saw no emission of light, and so we conclude that that sample did not have the DNA of our target species. We have, however, 2 samples where the DNA of the target species was detected, and we can see that with the fluorescence or the light that was emitted with each cycle of the PCR reaction. Now, I mentioned that this is quantitative in nature, and the difference between the 2 positive detections here is the amount of DNA. So if there's more DNA in the sample, you're going to see light emitted earlier, and that's what we see here versus in this sample. Now, instead of being able to say that there was just more DNA or less DNA, we can get much more specific and actually say there were, on average, 300 copies of bull trout DNA or lamprey DNA per liter of water that we sampled. And the way that we do that is we also analyze samples that we've spiked with a known quantity of DNA. And that ends up looking like this. So these are our spiked samples with a known quantity of DNA, and we can compare what their fluorescence looks like to our environmental sample, and that's how we estimate the amount of DNA in the sample. All right, so that's sort of the black box of what goes on in the lab, but how do we even develop these markers? We need species-specific markers. Well, what if you're interested in analyzing samples for a species that doesn't have a marker developed yet? There are two things that we consider when we develop markers, and that's the sensitivity of the assay, so the ability to detect all of your target species in the geographic range that you're interested in, and then the specificity, which is detecting only your species of interest and not getting false positive detections from closely related species. Our lab at the National Genomics Center has developed over 24 markers, and we have about 30 more that are in various stages of development. And through those processes of developing all these various markers, we've learned a lot of lessons, and we've come up with a checklist that we go through now when we're developing a marker. This checklist is a little bit long and a little bit technical, but the things I would like to emphasize are the first point, that we want to compile genetic data across the geographic range. And so that's going to include sequence data, which can also be available through some public online databases. Or sometimes we have to collect tissue and sequence the individuals ourselves. And then we screen the mitochondrial DNA region for unique targets. That is unique, to the target species. So an area of the genome that is specific to only our target species and is different for all other non-targets. So an area of the genome, for example, that all lamprey have in common and is not differentiated within lamprey, but will be different for other species. We then go through a few more processes, but we like to then test our candidate marker against DNA of both target and non-target species. And that's really critical because we want to make sure that our marker is performing when it's given quite a lot of DNA to work with. And we also want to make sure that if there's any diversity in the animals that we're surveying, whether they're targets or non-targets, that diversity that we haven't been able to see yet because maybe we haven't had sequence data available, that it's not going to result in unexpected results from our analyses. And then the last thing we do is we test against environmental samples with expected presence or absence. So an environmental sample that's a positive control and a negative control, again, to make sure that the assay is working as we expect it to. Even with this checklist, though, we still run into occasions where it's not perfect. So one example, focusing on the sensitivity of the eDNA marker or detecting all of your target species, this was a situation that we ran into when we were developing our marker for West Slope cutthroat trout. So this figure shows the development, or where we sourced tissues for West Slope cutthroat trout, Yellowstone cutthroat trout, and rainbow trout. And we tested tissue from over 30 West Slope cutthroat trout species across the range. And there was one individual in one population for which this marker would not work. And that was surprising because we worked really hard to be very inclusive with all of the samples that we were working with. But in the end, we looked a little bit closer and found that this particular individual, the genetic background of this particular individual was quite rare in the population, and we did not observe it in any other population. So the eDNA marker will still work for this population, but there are some fish that it's missing. So in this case, for example, trying to understand something like abundance would not be very accurate because you're going to have fish that are not being picked up with the marker, even though there are fish at the population level that are being detected. So that was an issue with sensitivity. An issue with specificity that we ran into was actually with our Pacific Lamprey marker. And so in the title here, a non-invasive tool to assess distributions of Pacific Lamprey, you'll note that we highlighted in the Columbia River Basin. And that's because, as you all know, Pacific Lamprey have a very different type of genetic substructuring compared to salmonids that have a lot of spatial substructuring because of their metapopulation dynamics. We found that it was very difficult to build a marker that would separate various Entosphenus species. So for this marker in the Columbia River Basin, where you know you only have Pacific Lamprey, this marker is going to work really well at picking up Pacific Lamprey. If you're in other areas where you have other Entosphenus species, this marker is also going to be useful for you. So for example, we've used this to detect Entus venus in the Klamath River Basin. However, if you have an area where you have multiple species of Entus venus present, this marker is not going to distinguish between those species. It's just going to say, yes, you have Entus venus present. All right, so now that we're kind of past that marker development and moving a little bit more towards understanding the application of eDNA, there are many papers addressing how does proximity, stream flow, water temperature, animal density, and other various environmental or biotic factors affect our detection probabilities. This is the type of thing where, again, I'm going to cover more in depth at Oregon AFS because there are many papers covering how these factors vary between water systems, between certain organisms that you might be trying to detect due to, say, varying life histories and that kind of thing. The long and short is that the closer you are to an animal, the more likely you are to detect it, and the more animals that are present, the more likely you are to detect them. And again, you can get a lot of publications that will address this in more detail from Taylor's website or just Google searches online. The take-home messages from all of these papers in general is that eDNA is a very powerful presence-absence tool. It's very good at telling you when animals are present and when they are not, even more so than traditional sampling methods. It is a somewhat useful tool for abundance, so it can tell you there are more animals in this location than there are in this other location. And there are some studies that have been able to really narrow that down in certain water bodies and say, okay, in this water body or under these environmental circumstances, we can get a fairly accurate estimate of abundance or biomass, but it's not easily extrapolated or applied broadly across watersheds. Environmental DNA is not a good tool to understand age or size structure. It's not a good tool to understand a population level of genetic diversity, and it's not going to be useful for understanding hybridization because we're looking in mitochondrial DNA, which is passed on maternally, and so that is just focusing on the maternal lineage. If you have a hybrid between a rainbow and a cutthroat trout, and the maternal lineage is cutthroat, you're going to detect cutthroat trout and not rainbow trout. So kind of moving on now into more of the application, how can we adjust protocols to maximize probability of detection? This is something that's really important for us to consider when thinking about lamprey because we have certain life stages for which the animal is not free swimming, right? During development, the amicetes are primarily in the sediment, so we don't really know how easily their DNA is being suspended. If we collect a water sample, how useful is that going to be for detecting lamprey in the system when adults aren't present? And we're going to hear a talk from Marty next in this session, or maybe I think third in this session, and she's going to address some of these questions, which I think will be really useful for all of us in this room thinking about how can we use eDNA when we're trying to address certain management questions for lamprey. So now kind of pushing on into the more recent publications, we've seen this explosion because we're at a point where we can start using environmental DNA to really get down to some ecological questions. So where are we finding animals and what environmental variables are associated with us finding them? We're worried less and less about some of these basic research questions because we've figured quite a lot out. It is still a little bit of an iterative process when we do these application projects. We still do end up learning quite a lot about the basic tool again, but we're able to start moving on. So environmental DNA applications for fisheries and conservation management. There are three major sort of facets in which environmental DNA is being used very effectively. The first is in eradication efforts. So this is usually when animals are in low abundance at the edge of their range in a given water body, and you want to make sure that you're getting, for example, all of the brook trout in this water system. So you want to know that your treatment area is fully encompassing that population. And then after you have a treatment, you want to make sure that you've gotten every single one of those fish. So when animals are in low abundance, eDNA is a great tool, and we've used it really effectively to monitor and assess eradication efforts. It's also being used in reintroduction efforts, and we're going to hear a little bit more about that from Carl. So watching animals move into an area as we take samples over time and see detections start to pop up where they didn't previously. And also using inventory and distribution, so understanding where animals are broadly across the landscape, covering broad swaths of land in a very efficient manner. Again, Carl is going to talk not only about reintroduction efforts, but also some distribution inventory sampling. So I look forward to hearing more about what Carl's been working on. And with my last remaining minute, I want to just give you a little bit of an overview of some of the research that's going on at the National Genomics Center where I work. One project that some of you may have heard about is our bull trout inventory study. It's a very large collaborative effort where we're trying to, after having identified suitable habitat for bull trout, which is based on climate shield prediction models, so areas that are cold enough to support bull trout, we're going through and actually sampling all of those areas to try and understand, are bull trout present in these areas where suitable habitat exists? We like to bring together big data sets, bring together a lot of partners to address these broad scale issues. So on that note, our Lampre research is kind of going into that same trajectory. We've been working on applications and development of markers to detect Lampetron and Dysphenis to again address issues of presence, distribution, monitoring populations, and then also recolonization. And what I've put here in the corner is a report that Ann Grote and I put together based on some reintroduction efforts in the Wenatchee and monitoring those, which was a really cool study to be a part of. We again learned a little bit about basic research. How do you sample a really big water system when you don't have a lot of fish and you want to maximize your probability of detection? Another project that's not exactly eDNA related, but is sort of starting to blend in with our other Lampre work is phylogenetic analysis to understand species relatedness across the landscape. And so, for example, we've been working closely with the Willamette National Forest to understand the diversity of both Lampetron and Dysphenis in their waterways, and that's been something that's kind of growing in a really exciting way. So in general, I'd like to give a shout out, a thank you to all of our colleagues and collaborators that have worked with us on some of these Lampre projects, and I'm really excited to see how these projects are all going to come together here today to help us better inform management and conservation for Lampre. So,
Great, round of applause.
that is all.
We probably have time for one or two quick questions.
All right, thanks. This is probably a naive question, but you said you could get the DNA signal from fecal samples, and I was wondering if any DNA from something that a fish ate could survive the digestive process and show up as DNA in a fecal sample.
Yeah, so that's something that, to the best of my knowledge, fecal DNA in fecal samples of fish has not been looked at, but one thing that has been looked at is DNA from predators. So is a predator, does fish DNA from a predator persist? And that's something that has been looked at, and in that case, yes, it does. So I would imagine the same would be true for fish. However, what fish are eating are probably also in the water that you'd be sampling anyway, so you wouldn't be able to tell if it came from fish species or another live individual.
Yeah, unless you had your predator moving between drainages.
Sure, so that's something that we've run into with various eradication efforts. We know we've eradicated brook trout from this area, but there are probably predators moving DNA because they're either walking, it's on their feet, or they've dropped a carcass, or they're defecating in the stream, and we're picking up that DNA signal. It's not indicative of a live fish, but it's indicative of DNA. And so honing in on sort of the local specifics of a given project is really important, and working with the biologists on the ground to understand the nuances of a given system.
You mentioned that the current PCR process was looking at single species, but you mentioned that also multi-species, and I've heard that that is happening. Is that close, do you think? Will we see eDNA sampling done on multiple species soon?
I'm not personally involved in the development of those projects, but I know there are a lot of labs, including ours, that are kind of sharing information and trying to get a multi-species approach up and running. It's something that we thought that 5 years ago we would be there now today, and so I don't want to say like, yes, it's going to be ready in 2 years. part of the difficulties with it are really technical, but it can be easy to those methods can be somewhat biased in that you might be only detecting the species that are most common, for example. And so trying to iron it out in a way where your results become unbiased is what's been really difficult. So I would like to say in another 5 years we'd be looking at that, but it's hard to know for sure.
Kelly. So next up we have Carl Busberg from USGS.
All right, so I'm going to talk about 3 Lampre eDNA field studies that I've been involved with, or that my group has been involved with. And the goal of this talk is not to rehash the same results over across these 3 studies, but to try to convey to you some of the things that we have learned about Lampre eDNA from these studies, and about Lampre that might kind of be interesting to everybody here. Okay, so the 3 talks that I'm going to talk about is first I'm going to talk about tracking colonization in the Elwha River following dam removal. I'm going to then talk about distribution and seasonal differences in eDNA across Puget Sound watersheds. Then I'm going to talk about spatial distribution and occupancy in the Chehalis River tributaries. But before I get any further, I just want to get everybody familiar with the eDNA method that we're using, and this is the method that's going to be applied across all of our studies, the studies that I'm going to be talking about here today. So we go out in the field, and can you hear me in the back? Okay, great. So we go out in the field and we collect 3 1-liter water samples at each site, okay? And that's represented here by the 3 1-liter cups here. And then we filter the water at those sites, and then we take those filters back to the lab where we extract DNA from each of those filters. And so you can see there's three tubes right up here, each from one of these filters, each representing one of the water samples. Then we perform qPCR in triplicate to test for the presence, or to test for the detection of our target species. So we get a total of 9 qPCRs going on from one site that we collect, so it ends up being 3 qPCRs from one water sample. There's 3 water samples, so we've got 9 total qPCRs that we're looking at. So the way I'm going to show the data here for this talk then is I'm going to show it in 2 different ways. I'm going to show the number of positive qPCRs in kind of it'll be a heat map that I'll be showing for the Elwha study, and it's going to go from obviously 0 to 9 positives, and you don't have to remember this calibration bar. I will be showing that on all the heat maps. The second way that we do it is through we quantify the DNA using standard curves, just like Kelly was talking about. Okay, so the first study then is using eDNA to track recolonization and dispersal of fishes following the removal of the dams on the Elwha. And so the removal of the 2 dams on the Elwha River is the largest project of its kind and is a significant restoration project for anadromous species. The Elwha Dam, the lower dam, was removed in April of 2012, and then the Glines Canyon Dam was removed in August of 2014. And so prior to the removal of these 2 dams, anadromous species were limited to the lower, were limited at 8 river kilometers below the Elwha Dam. There's no fish passage, when these dams were up. After the removal of these 2 dams, it opened up over 110 river kilometers for anadromous species. And so a key metric of recolonization of the Elwha by anadromous species is a key metric of success for the dam removal. So the Elwha, I'm going to be showing this schematic now. Here's a schematic of the Elwha. Here's a schematic of the Elwha River. Get out of your way, you can kind of see it. And I want everybody to be comfortable with this figure because I'll be showing this quite a bit. Step down, I guess, or something there for you guys, Ralph, if you can't see this. So the Elwha can be divided into the front country and the back country. Front country is easily accessible by car. There's a road that follows it. And so traditional field sampling methods are pretty easy to apply there. The back country is a totally different story. There's no roads. It's a roadless wilderness. And to get into the first part of the back country, it's a day hike. And to get any further, it's an extended overnight stays. And so traditional field sampling is really not very amenable to that back country area. So we thought that this is a great place to apply eDNA. We could use it back in the back country as well as in the front country too, but to track this recolonization. Okay, so the objective then for our study then on the LWA was to use spatial and temporal sampling strategy as a means for monitoring. Okay, so we're using eDNA to monitor the spatial extent of recolonization by the native species, and then also to monitor the dispersal of non-native brook trout that were introduced into the LWA between the two dams. So we're in our lab we're targeting we're using eDNA assays that target each of the 5 Pacific salmon, Omicus, the coastal cutthroat bull trout, Pacific Lamprey, and Lampetra, and then also the non-native brook trout. So, so here's the schematic. This is showing our spatial distribution of the LWA for our collection sites. So we've got 25 sites from the mouth of the LWA up pretty darn high, 60, 65 river kilometers or higher upstream. So we've got a really nice spatial stratification of sampling. These sites weren't chosen for any particular reason, or the locations that we're sampling. I think it's a little bit more ease of just kind of access to these sites. And then for our water collections then, we've, this table shows our temporal water sample collections. So any cell that's highlighted in this table, we've collected water samples from during that month. So we started collecting in 2014 in July, and that is 1 month before the Glines Canyon Dam was completely removed. And then, we've sampled all the way, just about every month up until, the month of December here. And for this talk, or for this portion of the LWA talk, I'm just going to be talking about the months from July 2014 through June of 2016. Okay? All right, so getting into the data then, what do the data look like? Here's a heat map of the complete data set from those from 2014 through June of 2016. Okay, so the things I want you to focus on here, the calibration scale bar down on the right-hand side, if you can see it. We're looking here at the number of positive qPCRs, so the darker the color in this heat map, the greater the number of positive qPCRs that we detected. So we've got the black, anything that's black is going to be a location that we didn't sample. So we haven't been sampling every location every time just because of logistics and difficulty of accessing some of these areas. Okay, so the first thing that I want to point out here for the LWA is that if you look at 2014 onward, and we're looking here at this boxed area in red, this is the lower river, the lower sample sites 1 through 4, what we see is that we get an increase in our detections over time, all right, as we move from 14 to 16. And this result totally jives with what survey and trap data are showing in the lower LWA, that Pacific Lamprey are increasing in abundance. They have been increasing abundance over time since 2014. Next thing here, what I want to show is that our detections are over time are progressing upstream. So if we look across the highest point in the drainage every year where we have multiple detections, or we have detections across multiple months within a year, this is what I've got circled here. All right, and so 2014 we get good detections in Indian Creek. Well, Indian Creek is a known hotspot for Pacific Lamprey. Their adults returning, they're reproducing, they're capturing larvae, they're capturing macrothalmia there. All right, 2015 we see that our detections have increased even higher up into the system, and we're getting replication across months, and this replication across months is really powerful, and we feel that this is really some really good results. It's great to have replication across months, and even 2016 also, it's increasing. It's going higher up into the system. So this is, I mean, we're really super pleased, and I think this is really cool, showing that these lamprey are stepping up into the system. Whether they're reproducing up here or what they're doing, we don't really know, but at least they're going up there and they're occupying that habitat. So one thing you'll notice here, there's a couple of sites here, here, and here where we have one detection. Okay? We have one detection across all of the months, and these sites are higher than any of the sites where we get multiple detections across months. So I've got them circled here. These sites right here in 2014 and 2016 that are circled in red, these represent one positive qPCR out of this whole 9 qPCRs that we did. So basically this is one qPCR was positive in one water sample that we collected, and the 2 other water samples we collected at that time were negative, okay? And you see the rest of the time it's negative across that month. Same thing for here. There is no detection. So we're exercising caution when interpreting these results, all right? We don't know. This could be a really low-level positive. Maybe there's a few individuals up there, and we're detecting it. Maybe it is a false positive. It's a little bit difficult to differentiate here, and I think this is something really important to understand if you're going to be using eDNA, or just when you're looking at talks on eDNA. This other sample that's up higher in 2016, this represents 3 positive qPCRs from one single water sample. So all of our qPCRs from one water sample, 1 out of the 3, was positive. And we feel that that's a bit of a much stronger result. It is replication. You get replication of qPCRs within that one water sample. Last thing I'm going to show about the LWA here is that our detections differ across months. So this is a really interesting result for the LWA. What I have boxed here in these red boxes, these are the winter months. It's kind of late fall, winter months here. And the unboxed areas are summer, spring and summer. And what you can see here is that in these boxes, within the red boxes, the detections are a lot lower. The color intensity is a lot less than the color intensity outside the boxes. We get a lot higher numbers of positive qPCRs in the spring and in the summer. This is really an interesting result. A couple of explanations for that result could be that water flow. All right, so higher water flows in the in the winter might be diluting out the eDNA that's in the system. Another explanation could be that in the spring and in the summer, you've got adults that are active. They're up in the water column, they're moving, and they're shedding eDNA into the water column. And so maybe that's what we're detecting. Okay, the second study I'm going to talk about is a study where we're looking at the distribution and seasonal differences in Pacific Lamprey and Lampetra species across 18 Puget Sound watersheds. And those 18 Puget Sound watersheds are shown here on the right-hand figure. And so the questions that we had was, how well does the eDNA reflect the known distribution of Pacific Lamprey and Lampetra? And are there seasonal differences in our ability to detect lamprey? We saw from the LWA that there appear to be some seasonal differences, but we wanted to see if these results could be replicated across multiple watersheds. So we performed our sampling in the fall, October to November, and we performed our sampling in the spring in May and June. So we went to each site in the fall, and each site then we returned in the spring, the following, that next spring. Okay, so the question then was, how well does the eDNA reflect the Pacific Lamprey distribution? So this, in this figure here, so we're just looking at Pacific Lamprey now. So in the figure here, at all of our sites, wherever you see there's black in this little half pie, that is where Pacific Lamprey are known to occur. And most of those data come from the study by Hayes et al. From 2013, where they went in and they visually identified or genetically identified individuals and species within each of these drainages. The eDNA results that we went back, that we collected, our eDNA results here then are shown in the orange in this slice of this pie. So, and then where Pacific Lamprey are not known to occur, or we didn't detect any eDNA, is in gray. And so what you can really see here is that we get 100, we get great, well, basically our eDNA replicated the results of the known distribution. So I mean, this is great news for us that, hey, we can go in and sample in these sites and get a pretty good representation of whether Pacific Lamprey are there or whether they're not. So we think that eDNA has a great power for these species. So the second question then was, are there seasonal differences in eDNA detection? Okay, so this figure here on the y-axis is the Pacific Lamprey eDNA mean copy number per liter. So that's how many copies there are per liter. Here we're quantifying our DNA here now. And all of these rivers here on the x-axis are rivers where Pacific Lamprey occurred that I just showed you in the previous figure. Okay, and so what we've got here is our fall sampling for eDNA is the orange bar, and our spring sampling for eDNA is the blue bar. And what stands out here is that there's way more blue bars than there are orange bars, right? So what that tells us is that out of the 14 sites that we detected Pacific Lamprey eDNA, we detected them every time in the spring, but we only detected Pacific Lamprey at 5 of these sites in the fall. Okay, so if we would then just sample in the fall, if that was the only time that we would have sampled, we would have gotten nearly 2/3 false negative results. All right, and that's assuming that larval Pacific Lamprey, or larvae are going to be present in these tributaries, in these streams year round. I mean, they're always going to be there. That's our assumption. Okay, and then the third and final study I'm going to talk about here is a study where we are assessing the spatial distribution and occupancy of larval lamprey in the Chehalis, in some Chehalis River tributaries. And so this study we collected. We went and sampled in the Black River, Skookumchuck River, the Newaukum River in 2015, and then we've sampled in the Wishkah and the Wainuchi River in 2017. And this is kind of a comparative study with eDNA and electrofishing also. Okay, so our objectives for this study then were to evaluate the performance of eDNA for detecting Pacific Lamprey and Lampetra compared to electrofishing, determine if eDNA quantity is distributed differently for Pacific Lamprey and Lampetra species within the tributaries, and then apply the eDNA to an occupancy modeling framework. We haven't gotten to the point of applying it to an occupancy model yet. We're going to wait until we get all our data collected from 2017. We haven't analyzed the 2017 data yet. So I'm going to talk about this 2015 collection that we did from the Black, Skookumchuck, and the Newaukum River. And so we've got 10 sites on each tributary, okay? And we have, we're targeting larval habitats, so type 1 substrate. Now we have patch sizes that are 50 meters, and these patches were determined by Fish and Wildlife using the grids method. So Fish and Wildlife went in and they electrofished in late September, early October in 2015. Then we went in and we collected eDNA samples late October 2015. So there's a couple of caveats to this study here I need to be up front with, and you got to understand. Number one, in an ideal world, we would have collected eDNA samples at the same time that we did the electrofishing. But unfortunately, it wasn't logistically possible at that time, so we had to wait a little bit, a couple of weeks afterwards. But we feel that it's not likely that a lot of larval lamprey would have been moving out, out of these patches between that time period when we electrofished and when we collected eDNA samples. The second caveat is that electrofishing is a great way to, if you want to know if lamprey are right there, you're going to go up and electrofish and you can get them to hand, you can see them. I mean, it's a great way to determine that a lamprey is right there in that specific patch that you're studying. The eDNA, if you're collecting at that patch, you're getting an influx from maybe that, from that patch, but also from any lamprey that might be above, okay? And you're also, if there's adults in the system, you're going to be getting eDNA off of those adults. But if you look at the big picture, if you just kind of don't get lost in those little weeds of the details of the details and the weeds, and you look at the big picture, it's just a general comparison between electrofishing and eDNA. I think it works out well. So for the results then, for that comparison between eDNA and electrofishing, at these sites where electrofishing and eDNA detected Pacific lamprey are represented in orange in this figure, okay? And where eDNA only detected Pacific Lamprey, there were 2 sites, there on the Black River and up here on the Skookumchuck River. There was 1 site where electrofishing only detected Pacific Lamprey. They weren't detected by eDNA. And then there were 2 sites where neither method detected Pacific Lamprey. So overall, I think there's pretty good agreement that eDNA does a pretty darn good job of looking at occupancy, or can be used for occupancy for Pacific Lamprey. It does a pretty nice job like electrofishing. And so the second question then is eDNA quantity distributed differently for Pacific Lamprey and Lampetra species within tributaries? So here on the y-axis, I've got eDNA copy number on the y-axis, okay? On the x-axis is the distance from the Chehalis River. The top graph is Pacific Lamprey from the Skookumchuck River, and Lampetra, bottom graph is Lampetra from the Skookumchuck River. And so the, boy, I just lost my thoughts here. So the distance then, as you're going down to the river mouth, the confluence with the Chehalis, you're going to the left of the graph. You're getting higher up in the system, you're going to the right of the graph. And so basically what we did then, we tested the hypothesis that we did a linear regression. We regressed eDNA copy number on the distance from the Chehalis River, that these are our patch size, or our patches. We regressed the distance of eDNA on those distance of patches to see if that slope of that line differed from 0. And it did, in fact, for Pacific Lamprey, there was a significant relationship here with eDNA copy number and distance. Okay, the eDNA copy number was higher, down lower in the system, and it dropped as it moved up higher into the system. That wasn't the case with Lampetra. Okay, the regression was non-significant, so this slope didn't differ from 0. So what these results might mean to me, or may suggest, we've got to be careful about over-interpreting some of these results, but it suggests that Pacific Lamprey, and this is really cool because it's a parent experiment from that same filter. We're sampling Pacific Lamprey and Lampetra, right? And we're sampling them from the same, we're testing them at that same patch site for each species. And so what these data may be suggest is that the eDNA copy number, it's higher, and the copy number is higher for Pacific Lamprey down lower in the system. It's lower as you move up in the system, and we know from a lot of studies eDNA abundance has been associated with the copy number and eDNA copy number and biomass. There's a relationship. So as biomass increases, there can be an increase in eDNA copy number. So these data might suggest that we have a higher abundance of Pacific Lamprey down lower in the system and a lower abundance of Pacific Lamprey in the upper system, whereas at these patch sites that we sampled, Lampetra doesn't may not differ in abundance as you move up. That's a bit hand-wavy, I realize that, but I think it opens a lot of questions and maybe some further studies to move forward on. So, summary, what are some of the lessons that we learned? That combined temporal and spatial sampling was a really powerful method to follow colonization and track colonization over time. We found that our detections increased over time by doing the spatial and temporal sampling. Single detections should be interpreted with caution. You need to be aware of those sorts of things. There were seasonal differences in eDNA detection. We had higher detection in the spring, lower detection in the fall. Stream flow could be an important component to that. Life history events may also be a really important component to that also. If you're detecting in the spring, you're getting a lot of adults and you're detecting those. And that's what's really driving that high detection in the spring. eDNA accurately identifies species distributions. eDNA performs comparably to electrofishing and for identifying occupancy, and eDNA concentrations, maybe we'll be able to use them to inform on the relative abundance and biomass of the species, but further studies are needed. And with that, that comes to the end.
All right, round of applause for Carl. Yes, we've got time for one or two questions.
Thanks for the talk, Carl. You had electrofishing data on the Chehalis. Were you able to correlate the upstream, downstream pattern of fish density with your hypothesis about the comparison with the EPA and the biomass?
That's the rub, and that's the rub, that we don't have any information on density, and so I think further studies would be really useful to do where we know relative densities at these different patches to see if that is indeed, if that holds through. But that's definitely the rub. I should have mentioned that.
I had seen a presentation on bull trout using eDNA, and one of the take-homes of that was that you had to be within 2 kilometers to get a signal from a bull trout. You had mentioned how close you think you'd need to be to the group of lamprey to get a signal.
You know, that's to be determined. We haven't done those sorts of studies. We haven't tested that so hard to say. I mean, we're getting these, we don't know how close we are to these lamprey. I mean, in the Chehalis system, we sampled in these patches, and we know from the Fish and Wildlife, from the services electrofishing, whether there were larvae there or not. But we don't know how well our eDNA collections are. If those larvae, how much do those larvae kick up into the water column? I mean, that's a big question. We don't know. So we don't know if there were any adults or how close we were to any adults.
I can maybe add to that a little bit. The 2 kilometers is, I think you're probably referring to some of the bull trout inventory stuff that Mike Young has been doing, and we believe that you can be about 1 to 2 kilometers away from a population density of bull trout to detect it, but it really starts to vary when you have like 1 fish. How close do you need to be? And I would guess that when you have free swimming adult lamprey, it's going to be probably fairly similar to what we've seen with different trout and salmon. But when you have, I mean, exactly what Carl is talking about, we don't know how much DNA is coming up when you only have ammocetes in the system, and that's something that needs to be explored.
I think we're going to have to hold further questions just until the break, or if we end up with a couple minutes at the end, because I want to give full time to Marty Lickey, who's our next speaker from USGS, who's going to be talking about sampling eDNA and sediments instead of water. And I'll flash 20 minutes for you.
Okay, thank you. Is anybody else feeling their lunch kick in and getting into that early afternoon sleepy? So if I start snoring in the middle, wake me up. So I'm going to shift gears a little bit, and like Ann said, we were interested in looking at eDNA in sediment. So that's not a common sampling approach for eDNA studies right now, but hopefully by the end of this talk I might be able to convince you that we think it might be interesting. Here's a general schematic. Oh, I'm going to have trouble with the microphone because I always want to turn. I'm going to try to do this. A general schematic that I think you probably already have in your head, but just kind of puts the pieces together. You have an aquatic animal, a fish in this little scenario, swimming around, and it's shedding DNA to the environment. I think that's what we've all been thinking about so far with eDNA. We know that once it's aqueous, floating around, it's going to be subject to this horizontal transport. Talk about sampling downstream, detecting things upstream of you, that's kind of logical. The piece that was new to me was this thought process of, depending on what packets, if you'll accept that term, what's being shed to the environment, whether that's gametes, talking about skin cells or feces, there's an aspect of settling, because all of those packets are pretty big, and eventually, depending on what the packet is, it's going to settle out. And when it settles out of the water column, it becomes sedimentary DNA. And then through natural mixing processes, I want to blind anybody with Bob's laser. Through natural processes, just wind, wave, tidal action, you get this resuspension, and that's what Carl was just speaking about too, that what's going on with the amicetes are things being kicked back up. So without doing much stretching of your imagination, we should be able to link kind of the opportunity to sample sediments with the thought process that what's in the water column will eventually end up down in the sediments. So when we sat down to think about Lampre, we wanted to look at sediments and wanted to tell you what we were thinking there. The thing I get when I read the literature is know your animal and know your system and figure that out before you start designing your eDNA study. And what we mean by that is a lot of the kinds of things you're hearing from Carl. Are the adults present? Do you expect gametes? Do you have decomposing carcasses? Do multiple life stages exist in the same time and space? Because when you get a hit from eDNA, you're not detecting a life stage or a given packet. You can't tell whether it's feces or a gamete. You're just saying we've got Pacific Lamprey. So you need to understand something about your system before you really set out to sample. So we decided we wanted to focus on amicetes. So right away, knowing your system means we should be looking at sediments if we're interested in amicetes. We were also interested because sediments tend to be less mobile, at least in some systems and for some sediments, but we thought that would give us an improved spatial linkage. So if I sample here and I get a detection, we're hoping that means the lamprey, at least the amicetes, are there. It can also be present for longer. There's some evidence that it has longer persistence. For example, this Turner paper in 2015, they sampled water and sediment and found that the eDNA in the sediment was present for longer than it was in the water. And then overall, we liked sediments because in the end, it's not limiting. You can still detect an adult lamprey or a MAC that was moving through the water column because of this settling aspect. So by sampling sediments, it doesn't mean you couldn't also detect those other life stages. So we knew we wanted to do amicetes, we knew we wanted to do sediment, and we sat down and said, what is it we're trying to learn? So we talked about the common questions that come up when you look at what inferences can you make with a positive eDNA detection. So take, for example, this little stream, and we're going to do an eDNA study with lamprey in this system. So we're going to go out and collect some water, and just for kicks, because it's a new part of our game, we're going to collect some sediment. We're going to take them all back to Carl's lab, and Carl and his staff are going to run a qPCR, because I don't do any of those things. And he's going to call and say, guess what, we detected lamprey in that sample. I said, okay, well, what does that mean? What did we just learn? Who was it that we detected? Well, we just said you're not going to be able to say that was an adult. Those are macrothamia moving through. Those were amicetes. All you can say is we got Pacific lamprey. How many are there? You got one fish, you got 1,000. We're not sure. Are they right here where I took my sample? Are they a kilometer away? Or are they 100 kilometers away? And are they there now? Or you think they were there and they're gone and we're still detecting that eDNA? So those are the questions that we took into our laboratory, and our idea was to set up controlled laboratory tests to try to see if we could put some sidebars on some of those questions that will limit our ability to make inference in the field. So what we did is we said for each question, how many amicetes are present? Well, let's run a biomass experiment. We'll look at the relationship between a known amount of fish, and then we'll determine how much eDNA we can get. This is kind of the holy grail. I think this is what all of us want from eDNA, is go out and get a water sample or sediment sample and say, oh, there's a ton of fish, or there's this many fish. Just because this is a beginning step, we decided we'd like to do this kind of categorically. There's a few fish, there's a middle range of fish, there's a high number of fish. So we didn't set out to do the kind of tight quantification, but more of a categorical approach. How far away? So we set up a distance experiment, and then how long ago might they have been there? So we set out to evaluate persistence. So those are the three pieces I want to talk to you about today. Brief touch on methods here. All this work was done at the USGS lab in Cook. We're using Pacific Lamprey amicetes that we raise in our laboratory. We did do some water sampling, but we're not going to present those data today. These were done in small tanks with some flowing water. because we raise fish at our facility, and you heard from both the previous talks, cross-contamination is a big concern, because over here in this lab, I'm rearing lamprey, and over here I want to make sure I'm only detecting the lamprey in the tanks. So we were very careful to test our water source and the lamprey food, and the sediment that the amicetes were burrowed in, to make sure we weren't introducing eDNA unintentionally. We used very clean sampling techniques with gloves and sterilizing sampling supplies, bleach to break down any eDNA we might have detected. Then we used the same assay that Carl was describing. In fact, Carl did all the assays. And in some cases, we got quantitative. We wanted to know how many copies. And in some cases, we were just looking at presence-absence. And that's what this slide tells you about. We used two different thresholds when we kind of looked at what our detections were. Sometimes we use the limit of detection, presence-absence, kind of a low bar. That's one copy of the DNA series we were looking at in a qPCR. So that's a low bar. Is there one copy there or not? Presence-absence. If we wanted to quantify, we used the limit of quantification, and that required kind of a higher bar. We needed 6 copies to get there, and we'll tell you when we're using either in the results here. All right, last little methods slide is the way we sampled our sediment. We did some testing early on to figure out, should we be scraping across the top, should we be plunging down into the sediment, and we concluded that we needed to kind of get down into the depth of the sediment a little bit, not just along the surface. And if you take a look at this slide, and this is the perimeter of the tank space, and you know there's amicetes in there, but we were using a different biomass levels, and we're saying, how do we sample this to make sure we're going to get our best odds of detection, knowing in the lab anyway that they're there. So we tried a couple different approaches and settled in on this one as it's a composite approach. So we sampled basically across the tank, kind of 12 different locations. At each sample, we put it into a clean container, mixed that all up as a composite sample across those 12, and then drew our samples out of that. And it's a way to kind of sample more space without processing each and every one of those samples. We think that we did some testing and convinced ourselves that improved our odds of detection. So all of our samples were collected using this kind of compositing approach. All right, biomass experiment. We tested 4 biomass levels, from 25 grams up to about 200 grams. The fish were about 80 millimeters and a gram each. So 25 grams of amicetes is kind of hard to wrap your head around, but lucky for us where they were all about a gram each. So in rough numbers, the 25 gram biomass level is about 25 individuals. We had 2 replicate tanks for everything, so this is the mean across those 2 tanks. So 100 grams was about 100 individuals, so just to give you a frame of reference. We put fish in the tanks, we gave them 14 days to kind of generate an eDNA signature, and then we did our sampling, and for this work we used our limit of quantification, because we wanted to know how much eDNA we were getting, not just were they there. Straight to the data set. On the y-axis, you have the mean copy number per gram of sediment, and across the x, you have each of the biomass levels. They're each represented twice, so you can see the difference between the replicate tanks. Note that at 25 grams of biomass, or about 25 fish, very, very low copy numbers, especially on this scale, it's difficult to see. And then our copy numbers went up with biomass. Then take a look at the variability between tanks. Pretty variable. At first we thought maybe we were not sampling effectively, but we did kind of a literature search backwards and through some of Carl's other sampling. Seems like there's more variability in eDNA detects than there might be in measuring 50 fish and looking at coefficient of variation for those. But there is this variability between one tank you get a hit, one tank you might not, or higher numbers or lower numbers. Okay, same data points, just in a scatter plot this time, and want to show you our regression relationship. So we have a slope significantly different than 0, and R-square of 0.77. So we have a pretty solid relationship between biomass and eDNA copy number. In theory, you could use this to estimate biomass if you go out in the field and get your sediment sample and come back and you say, I've got 150,000 copies, all right, that should be about 200 grams of fish. We just want to use this cautiously because we have a small range of biomass. It's not, it is a significant relationship, but it could use a few more samples in there before we want to use it quite that tightly. But I think it would be a powerful tool to say you've got a few fish or a big fish, a few fish in that area or a lot of fish in that area based on biomass. Trying to save you some time, maybe I'm moving too fast, but we're switching gears, moving on to persistence. So this is the second experiment. We basically took our biomass experiment and pulled the fish out and started monitoring through time. How long did the signal stay present? So the same biomass levels were used. In a general sense, our 25 grams was so low detection we didn't continue to monitor that. Our 100-gram tank, the 2 tanks there, we were using for water persistence, so we have data for sediment at 50 grams and 200 grams. One of the challenges we had was for ammocetes are burrowed in the sand, and we want to take the ammocetes out and leave the sand in the tank. And we didn't want to muck with things too much because we're afraid of kicking up eDNA and having it wash out of the tank with a flow process. So we used a mesh basket approach. That's what you see here is when we stock the tanks, the sand and the ammocetes are in the basket. When we got ready to start the persistence experiment, we kind of sieved the sand through, leaving the ammocetes in the basket. And that was a little disruptive in the short term. We turned off the water to the tank, tried to keep all that eDNA in there. And then we didn't sample for 7 days, hoping that things would settle. So we might be on the low end of persistence here if we washed out eDNA during that effort, but it was about the only way we could manage to get the ammocetes out and still leave material in the tank. We sample about once a week for up to a month. We dropped to monthly, and then in the end we were about every other month. And for this data set, we just looked at presence-absence using our limit of detection, and we didn't quantify. Okay, results. Different scale on the axis here. This is more akin to what Carl was presenting. The number of qPCR replicates above the level of detection, and the maximum number of replicates you could do is 9. So kind of 9 out of 9 would be the perfect score. Here again you see 2 bars representing the replicate tanks. The 50-gram biomass is in blue, and the 200-gram biomass is there in orange. So what did we find for how long these guys persist? We did really well at 50 grams out through 21 or 28 days. Then we went for a period of no detections, followed by a 1 replicate hit at 4 months. Carl was advising caution in interpreting, and that's kind of the way I read that record is, I'd like to find a way to validate that. So I think persistence kind of rounds out here, 21, 28 days at the 50-gram level. For 200 grams, more biomass, more persistence. We were pretty consistent here out to 4 months, although when you look closely again, you see that variability between tanks. Here you have one tank hitting at 3 qPCR replicates and the other one at 0. Here's a 4 qPCR and the paired tank at 0. So there's variability in this data set, but if you kind of take it in mass, we're about 4 months for 200 grams and about a month or so for 50. Okay, last experiment was looking at distance, and we settled in on using 100 grams as our biomass level for this experiment. We switched tanks and went to a long tank. This tank's about 5 meters long. We stocked fish into a similar kind of basket approach, put them here at one end of the tank, flow was moving down. We sampled that inside the basket, which was a positive control. We sampled at 0 distance, immediately downstream, and then about 1 meter, about 2 meters, and about 3 meters. Lisa Weiland and I, my co-author, were laughing at this morning because, why didn't you sample at 1 meter and 2 meter and 3 meters? Because we took a fractional approach, at least on paper. We said, let's take the distance and we'll sample at 25% of the difference. And then when we did the math, we're like, that was really silly. We should have sampled at exactly 1 meter, but that's just based on the distances in the tank. We sampled in the basket, and then at those distances, sampling from downstream first to avoid any contamination risk. Again, number of qPCR copies above the limit of detection. In this case, I've got my 2 replica tanks pooled, and the 2 bars are representing a day 14 sample and a day 28 sample. You'll see a positive control had perfect scores, so inside the basket where the fish were, we were able to detect them perfectly. However, if you literally move immediately downstream of the basket, your detections go way down. We certainly detected them, but the number of reps that got them was dramatically reduced. Similar finding at a meter, and these are 0 detects. So beyond that meter, we didn't get them. So somewhere between a meter and 2 meters, or 1.1 and 2.2 anyway, we lose our ability to detect at 100 grams biomass. Okay, wrapping this back up to our question we started off with, what does a eDNA detection mean? How many are there? Well, we have a relationship that when you do get a detection in the field, and if you can quantify it, then we could estimate your biomass at least within that with good confidence within the range we tested. We think we should use this kind of categorically. How far away are they? Less than 2 meters, because we got our detection at 1.1 and a 0 at the higher one. We think our ability to detect at distance is going to be related to biomass. That's kind of a common thread in the literature, and perhaps by sediment type, because we were testing in sand. Maybe if this was cobble or there was more room for water to flow through the material, we might improve our distance detection. How long? About a month or so at fairly low biomass, and out to 4 months was our last detection at high biomass. We didn't test at 5 months, and we had a negative at 6 months. So basically somewhere in that 4 to 5 months range is our last ability to detect that for persistence. So I like lab studies because you can control all your variables and you can really understand what's happening, but eventually we got to pull this stuff out into the field and see what that looks like when we put it on the ground. So if we want to sample this particular beach out on the Columbia, what might we consider based on what we've just learned in the lab? Do you want to sample sediment here, or water, or both? Well, that's not going to be an easy question to answer, but something you'll want to discuss, with your study team at the beginning. Part of that is how many samples can you process, how many technicians do you have, all of those kinds of questions. Something to think about though is if you're after recent occupancy, were they here, let's say you're doing something with contaminants, or if you want to know something about this last season, you might want to sample water because it will have less persistence than the sediment. If I was going to sample this stretch, I might draw out a transect and do sample points about every meter apart. If you're worried about low biomass, or if it's your determination of presence-absence is critical, you can close that distance down and hopefully increase your detection. The composite sampling approach lets you get more samples kind of through the qPCR without paying for them one for one. And what I mean by that is if you lay out a transect line, say it's 12 meters long, you're sampling every meter, so you come up with 12 subsamples. If you put them into a clean container and mix them up, now you can decide to take 5 subsamples out of that, or 4 subsamples, whatever you want to process, and it should represent that full transect because of the compositing approach. You should sample repeatedly over time. We learned that from Carl's data because things can change seasonally. Ask yourself whether you want to do quantification or just detection. That may drive some thought processes with how you sample. And then just a repeat note that you need to know your animal and your system. And Carl's data says that too, what his seasonal differences. You have to know something about what you expect to be in the area. If we were sampling on this beach and we were thinking ammocetes and sampling sediment, we'll have to remember that we could have adult eDNA contributing to that, which could obviously mess with your biomass relation, etc. So you have to know your system when you design and when you interpret. All right, last points are just where we might want to go with the data sets we have. We're in the process of getting the findings out for the sediment work, and it'll be combined with our water findings, so that'll be a neat story to tell. We'd like to see about using this tool now that it's got some sidebars in the field, maybe to evaluate impacts for dredging, or to look at water diversions. We talked earlier about the risk of dewatering. You might be able to get a handle on how many fish are there using some of our biomass pieces. And then generally improving our understanding of distribution. We'd also like to take this out in the field and do a specific validation piece similar to what Carl was discussing, where we did water versus electrofishing. We could expand that and say water sampling, sediment sampling, electrofishing, or some other direct sampling method to see how the lab pieces work in a real life setting in the field. And then anybody who's done lab work could probably sympathize with me that you start off with a set of questions and you think at the end you're going to answer those questions, and at the end you've just kind of got different questions. So we had some questions about what was going on with our sediment. Would that affect how far away or how long it lasted? Would that change if we had a different sediment type? We'd like to test additional biomass different levels, because we started at 25 and went to 200, but when we designed the work we had no information on whether those were going to be reasonable. So now that we have some data, we'd like to fill in that relationship. And then we'd like to test the relations between biomass and our ability to detect things, to detect eDNA at distance, because more animals is probably going to do better than a 1 meter detection if there's a bigger signal to begin with. And with that, I'll take any questions you may have.
Yeah, so we have a couple minutes for questions for Marty.
Well, or bring Carl and Kelly back up and we can do a bigger group too.
That works. Are there any specifically to Marty? And then I guess we can open it up to other folks.
Yeah.
Marty, great job.
Thanks, Marty. That was a great job. I was wondering, do you have any plans for testing the temperature? I'm thinking you're a cold-blooded animal, the higher metabolism, that would have higher shedding rates.
Yeah, I agree. Temperature, you know how it is with a pilot study. We don't have immediate plans, but I think temperature could definitely be a driver. We also had some questions about stress and whether the animals were, at some extra level of stress or were they calm. During one of our studies, we had some concerns about feeding lamprey in tanks can be, ammoseats in tanks can get to be messy because some of the residue builds up. We were wondering if that was messing with our detection ability, so we actually didn't feed the fish for some of the subsets of the experiments. And our concern was that stressful and that could change their shed rate, so we did a starvation trial on the side to make sure all of that. So there's certainly a lot more pieces that could drive this, temperature being a big one.
Other questions for Marty?
Yeah.
Have you thought of a way to differentiate the quantifying biomass and distance?
To me, they're not mutually exclusive. Like, if you get a big signal, does it mean there are more of them close, or are there a few of them far away? Or you see what I'm saying?
Yes, I definitely see what you're saying. Those are all things that we would need to refine from the work. I've not sat down and thought about how to tackle that, if that was your question, but could those two be confused? Yes, and we'd want to be able to tease that apart, and we're not there.
What about like turbidity? Like high water events versus like low water events? Like when you have high water events, there's more lamprey moving and there's more sediment being stirred up and more sediment moving down. If that changes like the read range, if you took when there's more sediment moving down and it's more disturbed and there's more lamprey moving through the system at that time.
You're asking what that might do to our ability to detect at distance?
Right, how that might change.
I'm sure that would have an influence. My guess would be that would potentially shorten your distance because things are so mobile. But I don't have any data on that, and that's one of the other reasons to take this and get it out on the ground in the field, because we had perfectly steady flow and perfectly steady temperature and kind of the dream conditions in a lab, and now it's time to see what that does in a field setting.
I know from experience that turbid waters fill up your filters really quickly, and so trying to filter 1 liter of water in a turbid system is difficult.
It might be another time to sample sediment, right? To do them side by side.
Do we have any other kind of general EDNA questions for either Kelly or Carl? Now's your time to get all your eDNA.
For Carl. And I don't know, so you can answer this. In one of your slides, when you're looking at Lampre versus Pacific Lamprey, I think you had eDNA copies. The scales were totally different on the y-axis. Is that related to biomass in that system? Could be. We don't know anything, as far as I don't know anything about the biomass, and that was a Skookumchuck River that we were looking at, so I don't know what the biomasses are like there.
The electrofishing show you the same result?
Well, the electrofishing, basically when they were electrofishing, they just started at the bottom of the patch, fished up until they were able to identify one individual from one species. So it was just presence absence. And so there wasn't any quantification or estimating any biomasses. But that would definitely be a follow-up. I mean, it's really great to hear all these questions that people have, because everybody that's asked a question here is basically a follow-up study that needs to be done. I mean, honestly, so these are really great questions.
Carl, I was just wondering, how much did it cost for the survey at OWA to process those samples for a full pass?
That's a, let me get back to you. Yeah, I mean, that's been, the LWA project has been funded from USGS yearly funds, and I don't know how much that those funds have been contributing to that analysis.
If I want to do a survey, how much do I need to budget?
Well, it depends on how many samples you want to do, what your question is, what your species, how many species you're doing.
Just assume that we're using your equipment.
All right. Doing that. Boy, is this getting recorded?
The cost structure is at the National Genomics Center. It's about $75 per sample, and that gets you all the materials and supplies needed to collect the sample, as well as analysis for one species. And then after that point, we have the DNA extracted, and it's about $25 per additional species analysis. So that's sort of our basic cost structure. But as Marty and Carl and I were kind of talking over lunch, when you are forming a project, it's really important to talk with the lab that you're working with, and that's going to allow you to think about, okay, how do I collect these samples in a manner that's going to be the most effective? We've had people collect samples, maybe oversampling where they didn't need to, or sampling in locations where we already have existing samples. And so as you're thinking about that and thinking about cost structure, have a good conversation with the lab that you're working with to figure out study design.
One more back here.
Perfect.
I just wanted to ask, seem like we're always putting numbers on animals and different animals, whether they be, buffalo fish or lamprey, and making a determination on how much should be here or how much should be there, and or this can only hold, and making a determination of saying this can only hold this much, when Mother Nature, had a vast area to have this, where the animals were put here on this world. It was only man that started determining the numbers that should be here for their benefit. And I don't think that that's something you should really check into on making a determination or putting numbers up, where they don't be putting a number on so many little things, and then making that determination that this is how this science is working, or what, that's what always bothers me and questions when you start putting numbers. It's the same way with salmon. You go in there where there used to be rows and rows and rows of beds of spawning salmon. Then biologists come in and say, oh, this can only hold 150 fish or 100 fish, when it used to hold 100,000. So that's what kind of bothers me when you start digging into this DNA sampling, and pretty soon is that going to be making a determination of there should be only 100 eels here, or there should be only 50 eels here, or 20 eels, or like that. That's what I don't understand.
Thank you for your comment. As far as I know, there aren't any plans to use a relationship of eDNA to biomass to in any way direct management that would be limiting lamprey. I think more than anything, we're trying to use this as a tool to identify places where we would like to have lamprey and maybe they're really rare. And we want to be able to prioritize those locations for translocation efforts or for habitat restoration efforts. And this is one of the tools in a whole toolbox that we can use to figure out, okay, what do we have for lamprey on the ground, and so how can we best restore them to places that they're absent and we would like them to be.
10 blocks more to the...
So with that, I think we're probably out of time for this session. I don't know what we're doing for break, but I did want to throw up really fast.
So anyway,
after
the last speaker's over, everybody's welcome to come over until the tab is full.
Put together a resource list.
Broadway Grill, 17th and Broadway.
But if you have additional eDNA questions, this is a list of all people who are working specifically.
All right, I've only got about
with
35 or 40
eDNA,
minutes worth of
so
some
feel free
introductory
to take
remarks here,
questions
and then we'll go
or-
ahead and start with the presentations. But I'm delighted to be able to facilitate this particular discussion. I think many of you know that the Yakima Nation was very interested in artificial propagation years ago, along with some of the members of CRITFC staff, particularly Bob Heinef, for those that you remember. And we started talking about artificial propagation really seriously in April of 2011, where we had the first international artificial propagation symposium, restoration and recovery symposium, here in the CRITFC building in 2011. And we brought over 4 researchers from Japan and some researchers out of Finland, if I remember correctly. And those folks had a lot of practical experience and successes to be able to share with folks at that symposium. And from there, we really did take off with a great start. And so I think what you'll see today in this discussion is some of that early information that Mary will be able to present to you, Mary Mosier. We've got Ron Feigl from the USGS, the Abernathy Lab, who will be speaking about nutrition. You're going to be rearing fish, you're going to need to be feeding them, and you need to be feeding them the right thing. And we didn't really know anything about what they were eating a few years back, so know quite a bit more now. And then our very own Ralph Lampman, who has been propagating fish in the Yakima Nation facilities at Prosser now for the last few years. And he'll be talking about where we're going to go from here. So you'll see a bit of a progression through these 3 presentations about how we got started, what we've learned, and where we're going to be able to go. And I'm going to ask the speakers to kind of keep a brisk clip, if you will, because I think where do we go from here is really a very interesting and an open question that I think deserves a little bit of time before folks take off this evening. One quick last note, if I can. This has been a great cooperation of funding, not only through the BPA and the U.S. Fish and Wildlife, but also Steve Hemstrom, are you still here? With Chelan County PUD, did Mr. Hemstrom? There's funds through Chelan County PUD that has also been very valuable to be able to help out with this effort, and so it's been a very robust joint effort by the Umatilla tribes, the Yakima Nation, and Mary's involvement with Umatilla and with NOAA, along with the funding from BPA. So enough said about that. Let me introduce to you Mary Mosier, please.
I love that, the words brisk clip out of the lips of Bob Rose. All right.
Come on, Mary, let's hurry up.
So I'm going to start out in sort of the beginning of this evolution of development of artificial propagation methods for Pacific Lamprey, and then Ron will follow on with sort of that next phase, and Ralph will finish this up. So we started in 2012, and I think one of the key things that'll come through all the way is that this has been done very collaboratively, and that has been incredibly important. We have very limited number of brood stock, and so we need to share expertise, we need to share facilities, and I really wanted to give a great shout out to Ralph because he and I really early on and starting in 2012 worked together in developing these methods along with teams from a lot of different agencies. So the brief structure of my talk is going to be some of the things we worked on at the very beginning, which is brood collection and sexual maturation of adults, fertilization and incubation of the eggs, and then some of the disinfection and transport pitfalls and successes we've had, and then I'll quickly touch on some little additional studies we did. So first, brood collection and sexual maturation. Here's a nice pregnant female. And typically the way this has been done is the brood stock are collected at the main stem dams in the summer before we are doing the artificial propagation with them. So they're collected and then overwintered at the various facilities. For the Umatilla tribe, we do it at the Minthorn holding facility through the winter, and then every one of those fish is sampled for genetics, and there's also a grab sample that's taken for disease testing that's done by the Oregon Department of Fish and Wildlife Fish Health Lab. And fortunately, we've never had any positive disease incidents for those grab samples. The genetic sampling has been super interesting on a lot of different levels. We look at the fish at different segments of their maturation, and then John Hess with CRITFC has been super collaborative in looking at the genetic samples, and we've used them for genomic studies. He also early on did some genetic work to confirm that we could get parentage assignments at a very high rate with fish that we had, artificially propagated fish that we have larvae from, that he could test back to those adults. So that was really a cool thing. So brood stock maturation, there's not a lot known about fish in the wild and how this process works. And we've learned a lot about it in the course of holding fish in the laboratory, or in these field settings also. The fish start to show final maturation at about 14 degrees centigrade at the Minthorn Springs holding facility. This is just temperature in this hazy blue line going up, and the date on the x-axis. So we knew that temperature was important. This has been also pretty well documented in the literature, but we didn't really know that much about how light and photoperiod might affect lamprey maturation. So we did experiments where we held broodstock in 3 different light treatments. One was darkness, which is kind of the control. The fins were the ones that originally said, oh, you got to hold them in darkness all through the winter so you don't, so they don't expend any energy and die from stress of being exposed to light. But we were worried that that might be messing with their maturation process, so we did some tanks where we had plexiglass covers on them to see whether that would affect both the synchrony of maturation and also the percentage of fish that matured. And then we also had an artificial 12-12 light cycle. And the data that we collected from this was just, we did a monthly sampling of just the fish that were clearly mature on the basis of their secondary sexual characteristics. So any of you who aren't in the know, the top one is a female. You notice the delicate feminine head there. And the bottom one is the male with a big fat head.
That's harassment.
So these are our data. We sampled in April, May, and June. And the first column are the dark treatment. The second column is the artificial 12-12, and the natural lighting is the third column. And the the males, the mature males are shown by the dark blue part of the pie, and the mature females are the peach color. And as you can see, there weren't huge effects of the lighting treatment. One of the things that's been troublesome is that we only get about 50% of our brood maturing, and we were hoping that this light treatment would get us past that, but in fact, we had almost identical final maturation rates for all three of the treatments. One thing that was interesting, though, is that the fish in the dark matured earlier than, for both sexes, than either of the light treatments. On to fertilization and incubation. Ralph and I, early on, we used to have spawning parties where we would meet up at one of our facilities and work on different methods for achieving higher incubation and fertilization success. And all of this was done, this is a lot of years of experiments, tons of information. I can't possibly touch on it all today, so I just wanted to hit some of the high points. And one of the things that's most interesting about this to me is it really has the potential to inform our understanding about wild fish, because we've learned so much just in having these fish in the lab and working with them. For one, repeat spawning in both sexes. You can hand strip a female, put her back in the tank, and hand strip her again a few days later and get another batch of eggs. I think this is a good indication that there's repeat spawning in the wild, and some of the genetic work that's been done recently sort of confirms that. The eggs are viable from freshly dead females, and we also are able to hold both eggs and milt for better than 24 hours, particularly if we put them on oxygen. So this gives us much greater flexibility when we have limited brood stock and we want to increase the genetic contribution of individuals. So that was an important finding. We also learned that they require very short contact times, and I guess this is sort of a no-brainer when you think about lamprey spawning in the wild in a flowing stream, that the eggs and milt are really not going to be in contact for very long. But we actually found that leaving them in contact for longer had negative consequences, so we only need to have them in contact for about 30 seconds. And then they're also very sensitive to physical damage, so you can't have a lot of tumbling. You have to rinse them very carefully so that they don't get damaged in that process. And Ralph did some really great work early on to reduce egg adhesion, because the eggs are incredibly adhesive as soon as they harden, and so they stick to the bowls, to your fingers, to everything in the lab, and they stick to each other, and this can create problems with fungal infection later on. So Ralph developed a method with pineapple juice where you can treat the eggs with 1% solution of pineapple juice to kind of reduce those adhesive properties. And finally, egg disinfection. This is something that salmonid culture folks know about for a long time, but we didn't really know what disinfection methods would be most useful with lamprey. And what we learned is that they're very sensitive, except for the very earliest egg stages. Iodophore is a killer for larval lamprey, but formalin seems to be a good disinfection method. And I know I breezed through all that, but it is encapsulated in this cookbook of lamprey culture that Ralph put together and published in Jawless Fishes of the World book. So that would be a really great resource if anybody's interested in the kind of nuts and bolts of that early fertilization and incubation process. As I mentioned, we learned a lot about how lamprey might incubate their eggs in the field from this work. And one of the things we learned is they do not need any flow. You can incubate eggs in an absolutely no flow setting with 100% success. And we did controlled experiments. This is percent fertilization on the left, percent development on the right to hatching. And the black bars are flowing setup. The white bars are just in a beaker with no air, nothing, just letting them go for a couple weeks, and we get essentially the same results. One of the other things we've learned a lot about is incubation timing. They're very sensitive to temperature, so any increase in the temperature shortens the incubation time. And we did some experiments with different water supplies, natural water supply, well water supply, and we didn't really get any difference in the incubation timing. But we did get differences between years. The blue bars here, the blue symbols are for the incubation mode for the first year we did this in 2015, and the pink is for 2016. And you can see there's a shift about 2 or 3 days, and we are not sure about, this is exactly the same temperature, same water supply, and so we're wondering about photoperiod possibly, or even possibly genetic determinant. And I wanted to touch on this. I mentioned that we had transportation and disinfection pitfalls. One of the pitfalls with disinfection is you don't want to expose them to iodophore too late in development. We also found that the early hatched, as soon as they hatch, they're very sensitive to vibration and also to changes in water quality. So Ralph did these really cool experiments up on the upper left, a vibration study where he exposed early hatchlings to high vibration for 5 hours and only had about 60% of them surviving. So, 60% of them were either dead or lethargic after that. So this has a lot of implications for the future. If people are going to be transporting larvae by FedEx, you might want to think about whether they're going to be jostled around a lot in the course of being transported. If you're going to be moving gametes or larvae between facilities, we need to keep these things in mind. And finally, I just wanted to touch on some of the other work. This is straying into the later evolution of artificial propagation that Ron will be talking about more of this stuff, but we have done experiments on temperature and salinity tolerance of these very early larvae, and they're incredibly tolerant of both rapid salinity changes and extremes of temperature up to 22 degrees and salinity up to 14 parts for short periods. The early larvae absolutely need to have substrate. We were hoping we could get away with no substrate because it obviously complicates culture to have mud in your lab, but unfortunately they absolutely need it to grow and survive. We did experiments on particle size of the food and learned that the particle size for these early fish is important because they are using their pharynx to strain the water, and those big particles may be too much for them. And finally, and Ralph's going to touch on this in much greater detail, but density affects obviously growth and survival, but lamprey are also amazingly resilient to surviving at high densities and surviving starvation for longer periods than Telias, and I think that's something that is really kind of a cool observation. So with that, I'm going to thank a whole raft of people that helped out with this, and I'm just going to flash up this slide because if anybody wants to see some cool lamprey spawning videos afterwards, I can show this. With that, I'll see. Any questions, I guess, or do I have time for questions?
No, yeah, we've got time for just a couple quick questions here. Ben, you got the microphone there.
Thank you. Any thoughts about how potential artificial pheromone plumes at these holding facilities may be attracting inadvertently adults from the wild and holding out below?
Yeah, it's a good question. Actually, in our facilities, I failed to mention that this work was done for the Umatilla tribe. We did it at the Walla Walla Community College Water and Environment Center and also at the Mukilteo Research Station north of Seattle, those two labs. And in both cases, our water goes into the sanitary sewer, and then out into Puget Sound in one case and onto a farm field in the other case. So we don't really have data from that, but Ron Twyble might be able to speak to that because their effluent goes into a stream, I think. So.
Another question? All right, thanks, Mary, very much.
Thanks.
Ron, you ready? Pleasure to introduce to you Mr. Ron Twyble from the U.S. Fish and Wildlife Service Abernathy Technology Center. Let's see, I think we're right here. Look familiar? Yes. And I think, where do you go? Oops.
USA Body.
Yeah, come here.
Make sure you click out of the old presentation in PowerPoint.
Well, good afternoon. Thank you for this opportunity to talk about our lamprey research at Abernathy Fish Technology Center. James Barron is our lamprey expert there at Abernathy, and he couldn't make it today. He has a prior engagement, so unfortunately you're stuck with me. My lamprey experience or expertise has to do with digging the lamprey out of the sand in the tanks that Mary alluded to. So I'm going to put up James's email on the last slide. If you have any questions about my talk or want to verify anything, send him an email. And so our research has been collaborative in nature with Mary at NOAA Fisheries and Ralph at the Yakima Nation. So the goals of our research have been to inform aniseed stocking decisions with regard to release locations in streams, and to learn more about culture techniques for this unique animal for purposes of research, for later release, for captive culture, and possibly for refugia. And also the ability to rear these animals in the lab may limit the need to perhaps mine wild populations for research. And so this is James here feeding his lamprey. And so some previous research we've done has included monitoring early larval development and survival, timing of first feeding, effects of water exchange rate on water quality and growth and survival of lamprey, effects of tank cleaning on water quality and growth and survival of the animals, even substrate grain size evaluations and their effects on growth of the aniseeds, and some temperature research as well. And so we've done a lot of nutrition work with these animals, looking at just the best type of feed to provide them, ration size, in conjunction with density, the effects of ration on water quality. We've even looked at vitamin and mineral supplementation to the diets fed to these animals. And James did a pretty interesting study looking at the use of steelhead effluent to rear these aniseeds in. And we've done some tagging work. Kyle Hansen published a paper recently on tagging of aniseeds. And we've done some screening work at Abernathy, and Doug Peterson is doing some work now looking at the effects of impact of irrigation diversion on lamprey. And so I'm going to focus on 3 studies very briefly that we conducted. One was a feed evaluation study, one was a timing of first feeding, and one was the rearing in steelhead hatchery effluent that I mentioned. So this is the experimental system that we've used for a lot of these studies. I think there are 40 tanks here. So this allows us to evaluate several treatments. It might be grain size for the sediment, or ration, or cleaning regimen, and use that on replicate numbers of tanks. We generally use three replicate tanks per treatment as a minimum. You can see the inflowing water here on this tank, an external standpipe, this banjo screen to prevent the lamprey from getting out of the tanks. You can see the sediment there. This is play sand that the aniseeds can burrow in, and then an air stone. So this is our typical setup for doing research there at Abernathy. And we also have this other room that James got set up where we can actually have different temperatures of flow through water. He bought these heaters that can actually heat the flow through water. And so we just finished a temperature study recently. These tanks are 38 liter in capacity. We use this play sand that we buy from a home supply store for the animals to burrow in, an effluent screen, and external standpipe. And so I'm going to talk about this feed evaluation study that we conducted. I think it was in 2012, and James has published this paper. So, of course, if we're going to hold these animals in captivity, we have to give them a good nutritionally complete feed, or at least try. So we evaluated 7 diets in this study. We use different ingredients. We use this active dry yeast, microalgae commercial blend, a mixture of yeast and algae. We even tried to use some fish oil paired with these first 3 diets. And we used an 80% yeast, 20% larval feed. And so each of these dietary treatments was fed to 3 replicate tanks of fish. I don't remember the numbers of fish that were stocked in each tank. It might have been 40 or 50. I don't recall now. But at the end of the 16 weeks, this was the result. So here are 5 representative animals from these triplicate tanks from each treatment. And so clearly the best growth was achieved when the fish were fed this 80% yeast, 20% LD, which is the larval diet. And so their length was significantly greater than that of all the other animals fed the other dietary treatments. And so the second study I'll talk about is this timing of first feeding. An important question for any species is when should we start feeding them? In the case of salmon, if you feed them too early, you can get a fungal development in the digestive tract that is not fully formed, and that can be bad. And of course, you don't want to wait too long to feed an animal too for its first feeding. So we conducted this study to see what would be the best time to start feeding them. And so feed was presented to the aniseeds at day 16, 24, 31, and 45 days post-hatch, or DPH. And so this is the result, survival. So survival was not significantly affected by the date of first feeding here. You can see it does tend to decline over time, but the p-value was not statistically significant here. But there was differences in growth based on when the fish were fed. So the lengths were greater or higher in fish that were fed on 16 or 24 days post-hatch compared with fish fed on 31 or 45 days post-hatch. And so this last experiment I'll talk about is this rearing and hatchery effluent. And this system here that James built, the blue tanks had steelhead stocked into them. I think there were 100 steelhead in the blue tanks, and then lamprey were stocked into those green tanks below. And there's no sediment in the green tanks in this photo, but we did put sediment in here in lamprey to determine whether can amicetes use effluent from fish culture. How will this food source affect lamprey body composition? We often look at body composition of animals in nutrition studies to get a general idea of their condition, and it's nice to compare the protein and lipid content of an animal's body compared with a control treatment. So that's what we did in this study. And will the amicetes sequester nutrients from the effluent and improve the water quality? So the lamprey in this study received either our control diet, this 80% yeast and 20% larval diet, or the steelhead tank effluent, or both. So the steelhead and the blue tanks, they were fed a salmon feed to, I think they were fed to satiation every day. So of course, the nutrients in the feces would wind up in the lamprey tanks for that treatment. Or the third treatment was the 80% yeast and 20% larval diet plus the effluent. So survival in all treatments at the end of the 9-week study was good. It was above 90%. And there were differences in growth. So the lamprey that received both the effluent and the control diet were significantly longer and significantly heavier than fish fed our control diet or the fish that received just the effluent. And we also saw some interesting results here with the body composition when we analyzed them for protein and lipid. What we noticed is that the protein content of the lamprey that received just the effluent was significantly lower than the lamprey that received our control diet or the control plus the effluent. And this could suggest that the protein was lacking that they received, which would sort of make sense because the steelhead utilized most of the protein in the feed, and so the feces doesn't contain a lot of protein, and that would be the source of nitrogen that the fish that received the effluent would have gotten. And also we saw a significant increase in the lipid content of the lamprey that got the control plus effluent diet. So compared with the lamprey that just received the control diet, that 5.8% there. So they probably just got more energy, is what I would assume here, and that would attribute to the higher levels of fat in those fish or lipid. So ongoing and future studies in collaboration with the ACOE Nation and NOAA Fisheries, and we're doing more nutrition work, more temperature work, some photoperiod work, and we continue to grow out cultured amicetes from brood year 2015. And so much of this research has been funded by my agency, U.S. Fish and Wildlife Service, as well as the Chelan PUD. And thank you, and if there's time, I'll try to answer a question.
Great stuff, Ryan. Thank you. Any questions here? We got time for a couple quick questions. All right. Thank you very much. So to conclude this, Ralph, you're going to have to show me which one of these. I think it's this PowerPoint right here. Yep. All right. When we don't often let Ralph out and amongst the public, but when we get him up behind a podium, we often find things out that we never knew. And I think that we're probably going to find a few things out here again. We hope that it has something to do with where we are now and where we're going to go from here. We'll see what happens. Let me get this. There you go.
you, everyone. Yep, my name is Ralph Lampre with the Accommodation Fisheries. Does this microphone work okay?
We can hook you up to the lapel mic if that would be helpful.
Just didn't want to block the...
Time's up.
Well said, Dave.
I want to emphasize this is a partnership project. We, with the Umatilla tribe, NOAA Fisheries, Fish and Wildlife Service, POR, BPA. I didn't put the Chelan PD funding. That's our recent funding I received. And so with that, oh, I was just kidding. Okay, so background, I'll talk a little bit about some of the background here. I want to talk a little bit about density studies, which we focus on space requirements for each life stage, and outplanting strategies that focus on the acclimation strategy and future plans. I want to, again, give the credit to the YKFP, lots of people, even Bob Rose, you can see him shoveling dirt that we use for the
Staged.
Yeah, he actually shoveled faster than me, I was looking at it. And we have this kind of the best management practice that really, it was a product of everyone, Mary, and everyone put as much work into this document. And there's a lot of, I think Mary mentioned this, but there's a lot of trials and errors. We try to do everything we can try with incubation, all the different methods that are out there, and kind of figure out what works, what doesn't work. With pro larvae, they're kind of unique. We get each female has about 100,000 eggs, so when we get them to hatch, we got to count them, and so we came up with, we came across expert count, which worked pretty good. We did have to put in some work to get it calibrated. And also started using Spawn Tech's mat, which is a coconut fiber, and that works really great for the pro larvae. They hide right underneath. So just before they're ready to burrow into the sediment, it gave them a place to hide, and that worked great. And Mary also mentioned this, but we tried going without the sediment, like trying the spawn mat to rear them into, but really they really have to have the sediment. It's kind of a given. That's kind of their nature, and that's where they survive. It's hard to take that away from them. Density this is kind of from the earlier results. We're seeing that if you pack them in too high, you're just not going to get as much growth. So there's kind of a threshold that we saw at about 100 grams per square meter, and that's with any size of larvae, small or large. When you pack in that much mass, then they just stop growing. So we did, with the Chelan PUD funding, we did some studies using our 20 aquarium. We looked at density, amount of feed, alternative feeds, methods of feeding. And usually after we rear them in the aquarium, we transfer it to the outside tanks, and this is one of our shallow troughs that we use for rearing. So general conclusion, so we put the density is about 3,000 per square meter if you convert it from aquarium. These are the 1,500, 750. We learned that definitely there's a difference. Low density, we saw the highest growth at 11 millimeter per month, and then gradually lower levels of growth for the high and medium density. But the next year we actually fed more feed for the medium and high density to see if that's going to compensate, and we did see now with the more feed we were able to bring that medium density growth to about the same level of low, and high density really raise it up as well. So there's still some compensation we can do. We're probably reaching the limits because the tank settings are getting really high with fungus, and whatnot, so I don't know if we can push any more, but we were able to get 180 to 200 grams per square meter, which is a lot higher than our initial threshold that we're seeing. Other conclusions were, mortality was highest in the early period, first 4 weeks, especially up to the second week. We tried, so there's some difference in how you feed them. So you can actually turn off the flow, and that really lets the yeast to settle down. And there are some risks of that, because if you forget to turn back the water on, you could kill almost all the fish in the tanks. And we were forgetful, so we didn't want to. We've tried that method, but it just had a lot of accidents. And so we've been using just a low amount of flow at the lowest level, and just keeping it at that. And with that method, we found feeding 3 times a week was better than 2 times a week. So we just tried with kind of two settings, but it may be different if you're stopping the flow. You may not want to feed 3 times a week. It just kind of depends on how you feed them. And we tried also feeding less amount at the beginning, because they're really small and maybe they don't need all that feed at the beginning, versus constant feed throughout the whole period. And we actually saw better growth and survival with constant feeding. So it seemed like even though their mass is pretty low, you still want to put all that feed in there just so they can grab what they need from that feed. And we've been experimenting with alternative feed, so like flour, wheat flour, dark rye, brown, and rice, or brown rice. We show that you can actually have pretty similar growth with a yeast in Otohime A1, or the larvae feed that Ron mentioned. You can get it at the similar levels. And also there's other alfalfa pellets and salmon carcass, or additional feeds that we add, and chicken eggs and leaves. All of these had really good, impressive impact on the growth, and really helped them grow. So survival rates initially, in the earlier years was only about 10 to 30%, really low. But in the recent years, we were able to get 72%. Last year, 85%, and this year is 93%, kind of our average survival in the tank. So every year it's been increasing. So initially, this is kind of the big picture of how much the survival at each stage. And we had a really kind of a bottleneck at that first feeding stage, which was low, but in recent years, we really brought that up to 90. So I think we could say we overcame that bottleneck, and we have a pretty high survival throughout. Alternative feeds, another reason we focus on this is we're pretty low budget, and active dry yeast, these early feeds can be pretty expensive. You can use these other things like leaves and salmon carcass if you have access through hatchery. It's pretty affordable or free. Flour, alfalfa pellets, chicken eggs are really, really cheap. But they're, they have the nutrition that really are found in streams, and I think they're great substitution. But another thing we have to worry is the space, what it takes to rear these larvae or adults. Adults really don't take much. You can pack them in. But then it gradually or exponentially increases. So by the time you have 1-year-old, to 4-year-old, you're going to have to really expand on your space. So, and we also see kind of a reduction in the growth kind of after the 6 months or so. So maybe that's a good time to get them out into the wild and or rear them in other areas. So just a kind of example with like 10 females, how many gametes that would be, about 1 million eggs. And if you have, looking at 50% survival, you're looking at 500,000 prolarvae. You can pack about 100,000 prolarvae in each of these troughs. It's about 16 foot by 1.5 feet. So you'll need 5 troughs, And if you have another 50% survival with 250,000 larvae, you can pack about 14,000 of these, and each of these long troughs are 14 feet by 3.1 feet. You'll need 17 tanks, and then if you're growing them to 1 year old, you can pack only about 800. That's if you're, like Mary said, you can really pack them in and they'll survive, but you're just not going to get very much high growth. So if you're really looking for growth, that's kind of the density you need to put them in. And so you can see you need 156, oops, I stopped. 156 tanks, at this level, so you really need a lot of space. So it makes sense to put them out in nature. Side channels have a lot of great habitat for larval lamprey, or acclimation ponds. We happen to have a lot in the Yakima Basin that have really good features for lamprey. Or canals, they do collect a lot of fine sediment. They have the dewatering issue, but if you can overcome that, there may be ways to utilize them. These are the VIE tags. You can tag young of the year fish with those, or pit tag them if they're 70 millimeter, or just use a parentage genetics to really track them. So we have a master plan that's about to be submitted, and in here, the Yakima Nation, these are the sites that we're going to focus on for initially, the 4 sites in the upper Yakima and 1 in the Cheese. These are the translocation streams that we primarily focus in the Yakima. So just going to give you kind of overview of the habitat area. We estimated habitat area, type 1, type 2, how much carrying capacity they have. And it's based on 10 grams per square meter, and that's kind of what we see in nature from electrofishing. That's kind of good sites provide about 10 grams, and hatchery setting is about 100 grams, but in nature it's a little lower. And type 2 is 1 gram, about 10% of the type 1 habitat. And we use kind of a mid-sized larvae, 80 millimeter, 1 gram, kind of the same size that Marty brought up earlier. And the carrying capacity we saw, like at Weenus, is about 50,000 larvae. So we were planning to put 3,000 2-year-olds, and it has a pit array, so we're hoping to get some out-migration data. And Holmes Acclimation Site is a little more habitat, a little more carrying capacity. We're planning to start on the lower reach first with 150 pro larvae, and then about 15,000 in the upper reach, and monitor them. Clallam is even bigger and even more carrying capacity, and plan to do a similar thing where you start on the bottom and then move our way up in the future releases. And then finally, Edgebuck Park, 2 releases. It does have beaver ponds and really good habitat. So future objectives, we want to continue to refine the hard prop incubation larval rearing. They can, hopefully we don't have to use it for conservation hatchery. Hopefully Lampre don't get to that point, but it will be useful in that aspect. But we also want to use our larvae for survival studies, with acoustic or pit tagging. We don't want to necessarily mine the wild fish as much. And then we want to monitor survival growth and behavior from our small-scale outplanting projects. I think it's a great way to learn more about their early life history biology. And in the long term, we can look at parentage assignments to see how many of the larvae released will survive. So yeah, with that, I want to emphasize that partnership is really what makes this possible, and then go into questions. And I have my contact info. I put together kind of a definition for supplementation, which is a little different from salmon. So lamprey, we're really focusing on we're trying to know more about the threats that are out there, and this is a tool for us to kind of learn more about those threats, and in addition to supplementing the population. And why, I think I'll leave this, let's just go to the questions, I think, from here. I brought up some questions here. I think it was James that brought these up. I can leave it in the background. Want to go to Q&A after this?
Yeah. Any quick questions for Ralph before we recap? And yeah, go ahead, Ben.
Question for Ralph and Brian. It wasn't clear. Sorry, I'm a slow
Why don't you come up here, Ron? So
question for Ralph and Brian. I wasn't clear. Have you guys released hatchery fish into the wild yet?
No, we haven't.
Okay, thanks.
Ben, yeah, we've got the document, probably in about the 99th percentile. We need to submit that to BPA, ISRP for review. Can't release any fish into the wild until we have that ISRP review and the go-ahead. And so that's what we'll be working on here in the next couple of years. Umatilla Tribe, Yakima Nation, along with Brian for the CRITFC, been developing that document. So we're right there on the edge of submitting that. And folks are more than welcome to read through that. And it provides, I think, good insights on how we anticipate moving forward with Lamprey supplementation, which includes both translocation and artificial propagation, and looking at the virtues, values, pros, and cons of each of these methods as we kind of move forward through the many numbers of years.
So, Ralph.
Oh, here we go. Okay, Steve Junkie.
Mr. Lamprey, ma'am. So your slide about you were having a mortality occurrence at that 3 months, were you able to determine what was causing that, those early years, what that mortality event was caused by?
No. They would just pop up on the top of the sediment and kind of have massive die-offs and kind of different periods in that early stage, and then when we actually sample the sediment, a lot of them were gone, and I don't have a clear. I think it relates to the feeding, that they're not getting the right feed at that early stage. Like Mary said, the really fine. Their mouth is really small, so it's got to be, the food's got to fit into that small mouth. And, maybe Mary, you want to expand on that? Yeah, and Ron, come on.
Turn that microphone off there.
Yeah.
Yeah, so I think this is something that's different from salmon, perhaps, is just that particle size seemed to be really important. And we did test. James Barron actually is the one who milled the feed down to less than 50 micron particles, and that was the feed size for those early pro-larvae that resulted in the highest growth, which sort of makes sense because they do, as Ralph mentioned, they can only handle small particles at those very early stages. So.
Ralph or Mary, there was a question Josh Moraskis had. Do you want to?
Yeah, I was just going to ask if, I don't know what the latest is on marking fish, so if you recover them later after release in the wild, where you can identify them?
Yeah, so definitely we'll be using, like I said, the VIE and pit tagging, and but there's also the parentage genetics. So from all the adults, we'll have genetics. So when we capture any fish in the wild, we can basically use the genetics to confirm if these are fish we release.
Yeah. John got like 98% assignment from what he did, and the ones that didn't assign were also kind of marginal DNA, so we're pretty happy with the ability to detect these hatchery reared fish from parentage
Ron, I was curious, do we have any sense of understanding how fish that are being fed in a hatchery environment might respond in a natural environment? Is there work being done on that or thoughts about that?
Not that I'm aware of.
Other questions?
As far as rearing, is the goal to put all the fish out to plantation or to rear some up to the macrophthalmia stage for research purposes? Is that something that you guys thought about possibly doing?
Yeah, the SLED funding was part of, one of the goals was so we can grow the macrophthalmia for survival studies, and that's definitely going to continue to be a goal for us. It is, you do need more space to grow them in large numbers, but yeah, that's definitely an important goal.
Yeah, if I could just reiterate on that too, Bob. You had the presentation this morning. We're finally getting a tag down to a useful size. If we can we now have enough macrophthalmia in our hatcheries, or anticipate having within the next year or two, so we can start putting these tags into these fish and running them through dams, running them through rivers, running them through these different environments, and being able to get good acoustic information, which is great information to have. So we've been envisioning this for quite a while now, and we really are just now on the thresholds of being able to have this capability. So it would be very interesting for all of you folks to be thinking about, where do we go from here? This is great technology, and we're finally to where we're hoping that we could be at. Any other response or insights? Kind of
When you release them, do you have any plan on like the depth of the water or any other parameters that will decide where exactly you're going to release them?
Well, I think the stage of development is really key, and I think Ralph did a good job of kind of talking about the plan for releasing at different life stages, because that would totally dictate that. These really early pro-larvae are not very mobile, and you would want to make sure you weren't just putting in a place where they're just going to get blasted out of there or eaten by predators. So yeah, I think it's all going to be really life stage dependent.
Yeah. There is, I think Umatilla plan is kind of focusing on that life stage specific difference, so they're really kind of putting smaller ones in this watershed versus that watershed, and we're kind of taking a different approach, and we're kind of putting them all in the same area to kind of see which life stage releases kind of you get the best survival. So kind of have different goals, but they're all important.
Another question?
I do, yeah, I do.
Oh, here we go.
Ralph, how did you come up with the 1% pineapple juice?
I was having a pina colada. It's in literature for different species. I'm forgetting the species now. So it was, we actually trying other methods of, there's other methods that have worked for other species, and pineapple juice, we didn't really think it would work, but I bought some on the side just to see if that would work, and all the other ones, a lot of the eggs just died from that treatment and didn't really work, and we had just one treatment with the pineapple juice, and that one had like all 100% survival, and they stick the eggs, and yeah, but I think it was the Fish and Wildlife Service that kind of brought that up initially.
Of course they did.
I might just mention one other thing that's a real interesting thing for folks to be thinking about. If you think about the propagation and you have the eggs in hand, which these folks can do very easily, fertilized eggs in hand, and you put those out into the river system wherever you choose, that's very, very cheap. And you can put, as was demonstrated, tens of thousands, millions of eggs out into the environment, versus holding them on to them for a month or 3 months or whatever the case would be, where you obviously are building up lots of expense. But what's interesting then is, so what happens to the eels once you put them into the environment, particularly at a very low cost, very young larval stage? And Josh asked the question about a marker on the fish. Well, all right, you put them in this area. Where do these fish go, if anywhere? Where do they die? And how would you ever actually be able to demonstrate that through a monitoring program? Can you actually mark these fish at such a small, and how big are they when you put the VIEs in there? 15 millimeters?
Yeah.
Something like that. So that's almost a year or half a year or so.
Almost like a couple of months or 1 month.
Oh, really? Okay, good. So it's really an interesting question about how to be able to monitor the fish and being able to understand how they're moving, if they're dying, these sorts of things. So there's a lot of interesting challenges, a lot of interesting creativity in our monitoring strategies as we go forward over the course of the next couple of years. And I just thought I'd put that plug in and folks can think about it. That creativity is kind of what we're looking for right now. Other questions? Keith Hatch.
At Abernathy, you had a slide with a range of temperatures. You didn't talk about kind of what the results were. I wondered what those patterns were.
That's because I forgot, and I had to send a text to James before my talk, and he said he thinks that we saw reduced growth at those higher temperatures, and I think 22 was the highest that he evaluated, and he thinks that we saw a reduction in growth. We just finished it, he's going through the data, and so that's what he could remember without having the data in front of him. Does that answer your question?
Yeah.
A reduction in growth at 22C, I think is what he said. But it'd be good to send him an email if you'd like, and you can get the data for sure. If you'd like to contact James Barron or me.
Christina, do you have another question back there? All right, great.
Hi, Bridget McConville, Warm Springs. My question is, are there any studies out there with natural occurring spawning and having the eels come back to where they were hatched from in their natural environment?
Like it says, are you talking about holding like the adults in the hatchery and then, oh no, you're just talking about?
No, those that come from the wild, and then is there any studies show that they come back to where they came from?
Yeah, the homing is pretty limited with lamprey, so you know, I think it was who was talking about that. Ben was talking about, you know, they may home in a large sense like Columbia River, but not necessarily in a stream sense, and so they don't necessarily come back to where, you know, where they were born from, and I think Aaron has looked at return from his initial.
Yeah, I don't know. He's got. I'd only add that there is this Statalyst project that CRITFC funded, and Lori Porter, I don't know if she's here, she might be able to address this, but I think they're actually going to be looking at perhaps that signal, homing signal in data from Lampre collected at Willamette Falls. But
And they're looking at, you know, we have our adults that we release this for translocation, and then, you know, about 10 years from now, some of those should be coming back. So, you know, we may be able to, if we're trapping them in our streams, we can collect the genetics and see if it's, you know, it relates to one of our parents that we had in our genetic database. So it'll take some time, 10 years or so, but we might be able to see that.
So just to talk about the Statalyst microchemistry analysis that Jessica Miller is doing at OSU. So she's looking at lamprey that were collected in the ocean, a set of lamprey from the ocean, a set from Willamette Falls, a set from Klamath River, and then looking at our larval lamprey from the Snake River Basin, the lower Granite Dam. And with the hope that there will be a signature that will show if some of the adults came from various basins, and then if and the adult signatures link back to the Snake River Basin, the Willamette River, and we're also looking for a marker in the ocean from entrance and exit of the ocean in order to start understanding their length of stay in the ocean, which is a great unknown. So she's only preliminarily looked at her data, and so far we don't have anything to report yet, but in about a year there will be.
This is a question pretty much for anyone out here. I was curious, once the larvae are kind of set up in the sediments and stuff, you know, get big high winter flows that moves the sediments and stuff around. Are the lamprey able to find new sediments to reestablish themselves, or are they kind of gone for good? I guess trying to think of like what's the limiting habitat variables in streams, and kind of using that to guide which life stage to start, you know, supplementing.
Well, I guess I can address it and then you can address it a little bit more. So the question about what are the limiting factors, I think the Finns actually tackled this early on because they didn't have a lot of spawning habitat, so they were interested in getting larvae out there. And one of the things they learned is that those really early pro-larvae potentially shelter in gravels as opposed to really fines. But then once they start burrowing, they need those fines in order to hold position. And they can get scoured out, but I think these guys will attest to the fact that when you roust them out, they're immediately able to get back into the sediment. I mean, they're much more athletic than you would imagine they could be. And so, you know, I think that concern is probably perhaps a little bit over emphasized, but they do have to have fines to be able to shelter. And I think Ralph's experiments with predation sort of show that level of it, that they have to have fines to also protect themselves from predation by teleos. So, you want to?
Yeah, I think the migration, you know, that you see in screw traps or other places, it's not just macrophthalmia, but larvae are migrating, but it's also, you know, whether it's active or passive, there's some debate there. I think they do migrate when, you know, turbidity is high because that protects them, and I have some kind of preliminary data from screw traps that show new moon phase. They were moving out actively on those phase, so they want the darkness, so maybe some of it's active and they're just trying to, maybe they ran out of, you know, they hit the critical threshold for density and they want to get out, look for more food and habitat and migrate downstream. And yeah.
Any more questions? One more question? Any more questions? I think you're just about warm out. Well, thanks. There's a lot of great work, and we certainly intend to be continuing to do this work over the years to come, so there'll be more information to share as we go. Thanks very, very much for the great work.
Okay, thank you so much, everybody, for coming today. We finished right on the button, 4:15. So Dave has already given you the info for the evening's activities. So hopefully a lot of you can meet over there tomorrow, and it's the Broadway Grill.
Broadway Grill. Let's all be Lampre nerds. Wear your name tag so it's a true networking event.
Good idea. And then you can put a hash mark when someone has had their paid-for beverage, right? No, thank you very much, everybody, for coming, and for all the speakers, and for all the good information. We will do this again tomorrow, starting at 9 tomorrow, not 8:30. So you can come here at 8:30 and meet and greet, but the meeting will start at 9 and finish at noon tomorrow. But hopefully we will see you tonight. Thank you.